जल प्रवाह पावितस्थले कलेवलं व्यलम्बितां भुजङ्गभङ्गमालिकामड्डमड्डमन्निनादवड्डमर्वयं चकार चण्डताण्डवं तनोतु नः शिवं शिवं शिवं
Thank you, Priyadarshini. That was mind-blowing. I request you all to devote some time to watch the glimpses which showcase the remarkable achievement of our institution. Moving on to the next video of Arham 360. Arham 360 is a revolutionary initiative founded by Executive Director of Sri Shanmugha Group of Institutions, Tirumurthy Arumugam, dedicated to bridging the gap between industry needs and academic curriculum. We empower students, institutions, and companies to thrive in a collaborative ecosystem that fosters innovation and talent development. It also offers a unique workspace environment designed to facilitate interaction and collaboration. The idea of Arham 360 emphasizes industry education, incubation, innovation, and internship.
Open your eyes. In a world where innovation and growth are key to success, businesses and young professionals face countless challenges. From finding the right resources to building their careers, the journey to success can be difficult and daunting. But what if there was a solution? We have created a space where innovation thrives, where the brightest minds come together to tackle the toughest challenges. We offer incubation programs that provide young professionals with the opportunity to gain valuable experience in their respective industries. Our experienced mentors guide and support them every step of the way, from manufacturing to IT and beyond. We're focused on driving progress across every industry. Our connections with top-tier institutions provide businesses with the resources they need to succeed. We believe that every idea is worth pursuing, no matter how big or small it is. That's why we foster a culture of creativity and collaboration where young professionals and businesses come together to turn ideas into reality. Our company is a representation of our core values, the 5 I's that guide everything we do: industry, institution, incubation, innovation, internship. We brought together some of the best and brightest minds in the industry, along with our key partners, to collaborate and drive innovation forward. Introducing Arham 360, a company that offers a dynamic space for innovative ideas and business growth, where we believe that success is not just a goal, it's a way of life.
Now, moving on to the next video, Inspire. Inspire is our skill-oriented flagship program for continuous 23 days. It helps students to acquire knowledge on industries other than academics.
In the heart of academia, where dreams are born and futures are forged, there exists a place of transformation, a place known as Inspire 23. For 23 unforgettable days, 68 extraordinary souls gathered here, not merely to learn but to transcend, not just to exist but to thrive. They came from different walks of life, each with their own story to tell, and what they found within these walls was beyond their wildest imaginations. This is the story of Inspire 23, a story of dreams taking flight where the ordinary becomes extraordinary. Inspire 23 is not just an event, it's a promise, a journey, a transformation. It's where students become leaders, learners become visionaries, and lives are forever altered. Inspire 23 is more than just a program, it's a promise of empowerment. Over 23 days, we nurtured holistic development among college students, equipping them with skills for life. Inaugurating the Inspire 23 program, we had the honor of Dr. V. Irayandu IAS, former Chief Secretary of the Government of Tamil Nadu. He has not only lit the ceremonial lamp but also ignited our spirits with a powerful motivational talk. The valedictory ceremony of Inspire 23 unfolded. It was a moment of pride as certificates were conferred upon our dedicated students, a testament to their hard work and commitment. Inspire 23 was not just a program, it was a journey of self-discovery and empowerment. From self-doubt to self-confidence, from uncertainty to purpose, these students discovered their true potential. As the sun sets on Inspire 23, our journey has only just begun. This is not the end, it's the beginning of a future where dreams take flight.
Thank you all for watching the video. Creativity is more than just being different. making the simple awesomely simple, that's creativity. It's with great pleasure and honor that I extend a very warm welcome to each and every one of you to the National Conference on Recent Innovations in Pharmacological Research 2024. As we gather today, we embark on a journey of knowledge, exchange, collaboration, and innovation that promises to shape the future of our field. This conference serves as a platform for experts, scholars, researchers, and practitioners from across the nation to converge, share insights, and explore new ideas. With a diverse range of presentations, workshops, and discussions lined up, I'm confident that this event will spark new ideas, foster meaningful connections, and drive progress in our collective pursuit of excellence. May I call upon Principal of Sri Shanmugha College of Pharmacy, Dr. P. Suresh Kumar, to deliver the welcome address. Sir, please.
A warm good morning to one and all. Respected Chairman of Sri Shanmugha Group of Institution, Thiru K. Shanmugam, Vice Chairman S. Prabhakar Sir, Executive Director and Founder of Arham 360, Mr. Tirumurthy Arham Sir, and Joint Secretary of Sri Shanmugha Group of Institution, Tirumadi Gokila Shanmugam Ma'am, and co-convener and vice principal, Sri Shanmugha College of Pharmacy, Dr. P. Perumal Sir. And chief guest, today our chief guest, Dr. K.S.G. Arulkumaran, professor and principal, KTN College of Pharmacy. I welcome you, sir. Thanks for our invitation. And also, I welcome Dr. N. Kannappan, professor and DRC member, Department of Pharmacy, Annamalai University. Welcome you, sir. And next, today our chairpersons, Dr. M. Surendra Kumar, Principal, Sengundar College of Pharmacy. I welcome you, sir. And Professor Dr. S. Ali Malarkodi, ma'am, Professor, Department of Pharmaceutics, the Erode College of Pharmacy. And also I welcome Dr. S. Rajarajan, Professor and HOD, KTN College of Pharmacy. And also I welcome our other peoples and head of all the departments and faculty members of Sri Shanmugha College of Pharmacy. The distinguished speakers and attendees, a very warm welcome to the 3 days National Conference on Recent Innovations in Pharmacological Research 2024, hosted by Sri Shanmugha College of Pharmacy, Sankari. As the principal of Sri Shanmugha College of Pharmacy, I am deeply honored to extend a heartfelt welcome to each one of you to this prestigious event. It is an absolute pleasure to see such a gathering of esteemed scholars, researchers, and professionals in our midst today. Your presence here today is a testament to your commitment to academic excellence and your dedication to the pursuit of knowledge. The incredibly proud and happy to be in front of you today as we gather to mark yet another significant milestone in the journey of our esteemed college. This conference serves as a platform for intellectual exchange, collaboration, and the sharing of knowledge, and I am delighted to witness to convergence of brilliant minds from various fields under one roof. Our institution has established a reputation for being a hub for intellectual achievements, shaping the minds of future generations and enabling them to achieve success. Our students routinely achieve academic success, exhibiting not only extraordinary knowledge but also a love of learning and creativity. Our college has established a reputation for being a hub for intellectual achievement, and shaping the minds of future generations, enabling them to achieve unprecedented success. As we gather here today, I am confident that this conference will serve as a platform for insightful discussions, fruitful collaborations, and innovative ideas that will shape the future of our respective fields. I encourage each of you to actively participate, engage in meaningful dialogue, and seize the opportunity to exchange perspectives with your peers. In closing, I would like to express my sincere gratitude to all the board members, participants, and organizers who have contributed to making this conference a reality. I am confident that together we will make this event a resounding success and pave the way for a future filled with innovations, discovery, and progress. So thank you once again for gracing us with your presence, and I wish you all a productive and enriching experience. Thank you very much.
Thank you, sir, for your wonderful welcoming. Our traditional auspicious lamp lighting as a tribute to Mother Saraswati, the goddess of knowledge. May the light of the lamp illuminate our path and guide us towards the truth and knowledge. Now I cordially invite our respective dignitaries to light the lamp. Now I kindly request all our dignitaries to remain on the stage. Now it's time to honor our dignitaries. May I request our Honorable Joint Secretary Tirumadi Kokila Shanmugam Ma'am to honor our Chief Guest Dr. K.S.G. Arulkumaran, M.Pharm, Ph.D. Professor cum Principal of KTN College of Pharmacy, Kerala, President, APSC Kerala Chapter, Secretary, ACPI South. Thank you, sir. May I request our Honorable Joint Secretary Ma'am to honor our Chief Guest Dr. N. Kannappan, M.Pharm, Ph.D., Professor and Drug Research Committee Member, Department of Pharmacy, Annamalai University, Tamil Nadu. Now, may I request our Honorable J.S. Ma'am to honor our speaker, Dr. J. Saravanan, Associate Professor, JSS College of Pharmacy, Ooty, Tamil Nadu. As we note forward with the inauguration ceremony, kindly request all guests on stage to take their seats in the audience area. Thank you for your cooperation.
Rhetoric is the art of ruling the mind of students. May I now invite Dr. C. Manigandan, Professor and Head of Department of Pharmacology, Shanmugha College of Pharmacy, to this 3 days national conference on recent innovations in pharmacological research 2024 to introduce our chief guest to this inauguration ceremony.
Very good morning to one and all. It's my pleasure to welcome our honorable chief guest, Dr. K.S.G. Arukumaran sir, Professor and Principal, KTN College of Pharmacy. Sir is President of Association of Pharmaceutical Science and Educators, and also Secretary of Association of Community Pharmacists of India. I welcome you, sir. So let's play the short video about for our chief guest, honorable.
I cordially welcome Dr. K.S.G. Arukumaran, M.Pharm, PhD, Professor cum Principal of KTN College of Pharmacy, Kerala, President APSC, Kerala Chapter, and Secretary of ACPI South, who has a wealth of experience and knowledge that will generously share with us today. Let's have an AV on our chief guest.
It's my honor to stand before you today and share the remarkable journey of Dr. K.S.G. Arukumaran, whose resume reflects not only a wealth of achievements but also a profound dedication to the field of academia and pharmacy. Dr. K.S.G. Arukumaran's career spans decades of unwavering commitment to education and professional development. As an esteemed academician, he has served in various capacities, from organizing secretary of prestigious events like Medifest 97, the Pharmacist 2005 National Level Sports Meet, to facilitating career opportunities for aspiring pharmacists at KMCH College of Pharmacy. His involvement in organizing national level seminars on pharmaceutical sciences and research and overseeing staff development programs underscores his passion for advancing knowledge and fostering growth within the pharmaceutical community. Beyond academia, Dr. K.S.G. Arukumaran has contributed significantly to professional ethics and standards, serving as a member of the Ethics Committee at KMCH Coimbatore. His role as an inspector for both the Pharmacy Council of India and the Tamil Nadu Dr. M.G.R. Medical University reflects his commitment to upholding quality assurance in pharmacy education. Furthermore, Dr. K.S.G. Arukumaran's active participation in editorial committees and scientific committees highlights his dedication to knowledge dissemination and research advancement. His contributions have not only shaped the academic landscape but have also inspired countless individuals to strive for greatness in their chosen field. Dr. K.S.G. Arukumaran's dedication to excellence in teaching has been recognized with the prestigious Best Teacher Award from the Tamil Nadu Dr. M.G.R. Medical University in 2011. Furthermore, his commitment to advancing pharmaceutical research has earned him the esteemed Research Excellence Award from Dr. Thanks Products, a GMP-certified herbal pharmaceutical manufacturer based in Tamil Nadu. This recognition highlights Dr. K.S.G. Arukumaran's significant contributions to the field of pharmacy, further cementing his reputation as a leading authority in the industry. I am honored to spotlight the remarkable academic achievements and recognitions earned by Dr. K.S.G. Arukumaran, a luminary in the field of pharmacy education and research. In addition to his professional accomplishments, Dr. Arukumaran's alma mater, the Sri Ramakrishna Institute of Paramedical Sciences in Coimbatore, bestowed upon him the esteemed Best Alumni Award. This recognition speaks volumes about his enduring connections to his alma mater and his exemplary achievements in the field of pharmacy. and accolades not only reflects his exceptional talent and dedication, but also serve as an inspiration to all aspiring pharmacists and educators. With a robust academic background and an extensive list of publications spanning decades, Dr. K.S.G. Arulkumaran has demonstrated an unwavering commitment to advancing pharmaceutical research and improving healthcare outcomes. His pioneering work in areas such as formulation development, drug utilization evaluation, and pharmacognosy has left an indelible mark on the field, shaping the way we understand and approach the patient treatment. Beyond his academic achievements, Dr. Arulkumaran's passion for improving patient education and safety is evident in his studies on the readability and effectiveness of patient information leaflets. By bridging the gap between complex medical information and patient comprehension, he has empowered individuals to take control of their health and make informed decisions and their treatment. Furthermore, Dr. K.S.G. Arulkumaran's exploration of natural binding agents for pharmaceutical formulations showcases his commitment to sustainability and harnessing the potential of botanical resources. His research not only opens new avenues for drug delivery but also underscores the importance of environment-friendly practices in pharmaceutical manufacturing. In addition to his scholarly pursuits, Dr. K.S.G. Arulkumaran has been a mentor and inspiration to countless students and researchers, nurturing the next generation of pharmaceutical scientists and instilling in them the values of integrity, diligence, and scientific rigor. Dr. K.S.G. Arulkumaran's contribution to the field of pharmaceutical sciences are nothing short of transformative. His tireless dedication to advancing knowledge, improving patient care, and fostering innovation serves as a testament to his unwavering commitment to the betterment of humanity. Please join me in welcoming and honoring Dr. K.S.G. Arulkumaran for his outstanding achievements and continued dedication to excellence in pharmaceutical research and patient care.
Now I kindly invite our chief guest, Dr. K.S.G. Arulkumaran, to the stage. Sir, please. Now I also would like to invite our principal, Dr. P. Suresh Kumar, to honor our chief guest.
I kindly invite Dr. K.S.G. Arulkumaran sir to share a few words.
I just melted by seeing this particular video, right? So I have reserved my gratitude to those who take this particular videograph. Just while you please let me hand over the video clips. I think it will be more useful for me. I can show to my family members. Ellorukkum vanakkam. At the very outset, I would like to thank all the officials of Shanmugha Group of Institutions for inviting me as a chief guest for the 3 days international conference on recent innovation in pharmacological research 2024. 2 days back, I just read an article. It says that, question mark, do you know who is the real man? The question is, do you know who is the real man? Any answer from the audience side? Who is the real man? So it is written there, the one who gives respect to women. So in this connection, so I thank Srimati Gogila Shanmugham for inviting me as chief guest, ma'am. Winners are ordinary people with extraordinary heart. May the bells of happiness always rings in your life, ma'am, dear ma'am. I give a big hands to our Kokila ma'am today. Gratitude is the best attitude. In this way, I thank principal of this great institution, Dr. P. Suresh Kumar, for inviting me again. And my colleagues, Dr. Kannappan, and many of the speakers over here. I'm sure you'll be getting a lot of terminology while going out of this institution. I also thank Dr. K. Surendra Kumar. I know he is having many fans over here. Generally, I used to say, I used to introduce my students to the audience. So he is my students. But Surendra Kumar sides, so I have to say, I am his teacher. So such a kind of person is Mr. Surendra Kumar. If one can do, anyone can do it. If no one can do, you have to do it. So in this connection, I thank my student, Dr. Gopal. Yeah, give a big hand. The man behind this 3 days seminar. Brave concept. You know the brave concept? Generally do or die. Practical concept, do it before die. The brave concept is do or die. Practical concept is do it before die. Our chairman Shanmugha sir concept, you know what is his concept? Don't die until you do it. So in this way, I also thank our chairman through Shanmugha sir for giving this excellent opportunity me to be a chief guest of today's function. So I thoroughly enjoyed the welcome note and all other speech over here. But my kind request is, so do you all like stories, my dear students? Say yes or no. So I have 5 stories with me, shall I? And the stories goes like this. In a fine morning, there is a village near to Erode, but there was no rain at all in this particular village. So they pray for God, right? So they are all assembled in one ground. They are all assembled in one ground at a particular time. 12 o'clock means 12 o'clock. One small boy came with an umbrella. That is called, which one? Hope. Hope or faith? Faith, right? I think the credit goes to Surendra Kumar, if I'm right. Yeah. So next story. This is a small story, right? So you should all understand, if you want to come or if you want to excel in your area where you are, so you have to follow all those things, what I'm trying to say now. The second story is, when you throw the babies into the air, they will not cry. Instead, they will laugh. You know what is the reason behind? They know that you will catch hold them, right? That is called what? Trust. Okay. So second story is over. Small story. Again, I am repeating you. Every night when you go to bed, right? So without any assurance of being alive, we set an alarm. Yes or no? Right? That is called what? Hope. Okay? And fourth one is, we plan big things for tomorrow without about the future. So May month we have this program, June month we have this program, but we don't know what will happen during those days. That is called hope. Okay? confidence. And when we see the world suffering, but still we get married and have children, that is called love. And fifth story is, an old man, his shirt was written, I am not 80 years old. I am sweet 16 plus 64 years of experience. That shows his attitude. So what all things I have narrated, you just let me know. So I'll be happy. I want to hear from you. What was the first one? Hope or faith? Second one, trust. Third one, actually first one is faith, second one is trust, then hope and confidence, love and attitude. To top up with, you all should have two more qualities, or I can say attitudes, that is adjustment and compromise. What are they? Adjustment and compromise. Adjust when someone wants with you, and compromise when you want to be with someone. So if you have all the characteristics, each one should have all those characteristics, you can excel whatever the field you are. That's what I wanted to this one inform you that you must be optimistic. And my dear students, when I was in Muscat, 3 years I was in Muscat, right? I had a wonderful time over there. So I'm telling the importance of communication, not only English, the other languages also. The pharmacy where I was working is a polypharmacy. There you could see one dentist, one GP, and a pharmacist. Dentist from Pakistan, GP, general practitioner is from Iraq, of course, pharmacist from India. So when we have a tie-up with the company, Bengali company over there, so these employees, they used to talk only in Hindi. So who were expatriate, they used to call us Hindi wala. Yes or no? So, but I don't know single word in Hindi those days. Now I learned something, I can speak something Hindi. Those days, zero knowledge about Hindi. So whenever they used to meet me, Hare bhai, kaisa bhai, TK, they started blah, 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 many things. I don't know single word in Hindi. So they will also understand. No, so he doesn't know Hindi and all those things. They approach the Pakistani doctor. Pakistani knows Hindi very well. So he explained to the GP doctors that particular person is suffering from diarrhea, or that particular person is suffering from headache, all those things. He narrated such a thing to the doctors. After 5 minutes, he called me, right, over phone, he called me, what is this, Arul? What you are doing? Are you from India or some other place, right? Being an Indian, you should know Hindi. See, Pakistani explained to me, right? He's translating many things to me, but you don't know how to talk in Hindi. I said, sir, I am not from India. Who said I am from India? I am from Sri Lanka. So I have to escape, right? So that's what communication is very, very important. So my dear students, you try to develop communication. Right? Communication means language. Try to learn many languages. That will help you out when you are in a difficult situation. One more thing. Also, and when I think just 1 week back, so one foreign Benz car came with beautiful girls. Omani girls are so looking good. So they also approached me. They used to talk only in Arabic. And how are you, right? Guess this one now. Salam alaikum. They used to start like this. Salam alaikum. Kef walak. Kef saka. Kef abu. Kef soja. Al zain, zain, zain. We have to say zain, 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 all those things. Then she asked me, Inta araf hada hamam. she asked me, Inta Araf had a hammam. Means, do you have a hammam? I said, hammam is not available here. Hammam minni mafi, Mawjood India. I said, hammam soap is not available in Oman. It is available only in India. அந்த பொண்ணோட ஃபேஸ் மாறி போயிடுச்சு. ஏதோ ஒரு படத்தில் வர மாரி ரெட் ஆயிடுச்சு. ஹர் ஃபேஸ் டென்ட் இன்டு ரெட் கலர். She fastly approached our GP doctor since he knows Arabic. And again after 5, Arul, you are always troubling me. Why you said like this? Sir, I didn't say anything, sir. I said, hammam is available, no? So, no, you have to say sorry to them. And I don't know what was happening. Immediately I called my friend over there. He knows Arabic. Then what was to happen? You just let me know. I narrated the thing. They came and they asked me the hammam. I said hammam is not available in India. Available in Oman. It is available in India. He also laughed. Oh, you will be in trouble. No, and I confused like anything. I said I have to admit sometimes mistake. Said this time, no, it is a fact. Hammam is available in India only. Yes or no? It may not be available in other countries. So he told me after some times, see, Arul, hammam is the bathroom key. What is that? Hammam is the, if you translate the hammam, it's a key of bathroom. They asked me the bathroom key. I said, bathroom is available in India, not available in. So they also sometimes, no, you should be, and the அவசரப்பட்டு பேசுகிறோம், அவசரப்பட்டு செய்யக்கூடாதுல்ல, நிதானம் உங்களுக்கு இருக்கு. You should have some patience. Counseling is also very, very important. My dear students, I think you, the Shanmugha College of Pharmacy also started PharmD courses. I could see some of the PharmD students over here also. So patient counseling is very, very important. Patient counseling is just like that. You are communicating the thing to the patients. Effective communication. Even you are dispensing paracetamol, you have to counsel the patient that paracetamol has its own disadvantages. Don't think that paracetamol is very safe. It has its own side effect. Even if you keep on taking paracetamol, you may land up with the kidney issue also sometimes. And if the dose goes above 5 grams, you have to face the consequences like liver toxicity. There is a overdose. This is N-acetyl system. You all know that, right? My dear B.Pharm students and PharmD students. That's what I am telling you. Your job is to counsel the patient, because doctors are there very busy with their own activity. You are the right professional, right healthcare professional to communicate to the students. If you are not communicating, what will happen? For example, I can say, one patient is coming to me. He is asking 5 ml, that one teaspoon plus CPM. Anyone, can you abbreviate what is CPM, my dear friends? Chlorpheniramine maleate. Yes. So, what is the action of CPM? Antihistamine. Yes. So I'm checking the presenter. He is good or not. Yes, credit goes to you, sir. It produces sleep. Obviously, CPM produces sleep. So if I am not communicating to the patient that you have to take 5 ml only of that particular CPM, what will happen? You think, just imagine, if you don't know the posology, right? If you say something, teaspoon, is in general you are telling 1 teaspoon for 3 times a day. That means the patient will go to his house, right? So if there is no teaspoon over there, he or she may use the sambar karandiyoda edutruvaanga. So think of that. What is the consequence if you are taking sambar full of CPM? So it is your duty, pharmacist's duty, to take care of the patients. Not only getting money is not in your job. Your primary job is to educate the patient. This is how you have to take. 3 times a day, 5 ml. Teaspoon, tablespoonful, dessert spoonful, wine glass fulls, tumbler full. These all some of the basic posology matter pharmacist, whether you are a diploma of pharmacy, you are a bachelor of pharmacy, master of pharmacy, even PharmD students. These are some of the basic things you should carry out and that patient should reach. And also, for example, assume that you are a pharmacist working a good pharmacist, patient is coming to you and he is asking suppositories. For example, suppositories. If you are not giving the proper educational, or I can say the counseling points to the patients, they may swallow the suppositories. What they supposed to do for suppositories, please let me know. They supposed to insert the suppositories into the rectum. It depends upon the thing. There are different dosage form, right? Conventional dosage form, sustained release dosage forms, delayed release dosage forms, and there are many terminologies used in us. Those terminology we cannot expect the layman to understand. If you are giving the sustained release dosage form to the patient without communicating the patients, what will happen? He or she may take the tab, one in the morning, one in the afternoon, one at night. What will happen? Just imagine. He or she has to face the many consequences. Actually, they are supposed to take one tablet for either 4 hours or depends. Doctor may tell you the right exact dosage of thing, right? And also, even while dispensing, there is a drug called atorvastatin. Hope you understand what is the use of atorvastatin. What is the use of atorvastatin? I need your response, my dear students. I'm happy that your great presence is very important. I think you are all physically present, mentally absent, I think, right? Going here and there, mind. You say something, then I can understand you are active. What is the use of atorvastatin? Which one? It reduces cholesterol. Yes. So while dispensing particular anti-static drugs, you supposed to educate the patient. So this is the cholesterol drug. You have to take this particular drug at night. Why? What is the reason? Why not to take in the morning or why not to take in the afternoon? The reason is cholesterol synthesis will happen only during night. If you are asking the patient or if you are not giving information to the patient may take the tablet at morning. So he will complain about the doctor is not good. எங்க MBBS படிச்சாருன்னு தெரியல, ஒரு யூஸ்மே இல்லைன்னு. Right? So shall I communicate in English or Tamil or both? Both. Okay. One more thing. ஒரு பேஷண்ட் வந்தாரு, obese patient, டாக்டர் பேசுறாரு, சார் ஐ வாண்ட் டு ரெடியூஸ் மை வெயிட். டாக்டர் சொன்னாரு, இமிடியட்லி சார் யூ கேன் ஹாவ் அ 2 round நேரு ஸ்டேடியத்தில் சொன்னார். 2 round he did. அப்புறம் 1 month கழிச்சு வந்தார், சார் வெயிட் இன்க்ரீஸ் ஆயிடுச்சு சார் 2 kgன்னு. வாட் இஸ் திஸ்? ஓகே, இன்ஸ்டெட் ஆஃப் 2 round why not to go for 4 rounds? அப்படியே டபுள் ஆக்கிட்டார். 4 rounds போயிட்டு வந்து திருப்பி 2 மாதம் கழிச்சு வந்தார், இன்னும் 2 கிலோ ஏறிடுச்சு. டாக்டரே கன்ஃபியூஸ் ஆகிட்டார். என்ன சரி இன்னுமே இவருக்கு ரொம்ப கொடுக்கணும் போடுறக்குன்னு சொல்லி சரி 10 round அடிங்க, 6 மாதத்துக்கு கழிச்சு வந்து பாருங்கன்னா அவர் 120 போயிட்டார். அப்புறம் டாக்டருக்கே சரி இதை நம்ம செக் பண்ணணும், சில இதெல்லாம் பண்ணணும்னு சொல்லிட்டு எத்தனை மணிக்கு போவீங்க சார்னு கேட்டார். நான் 6 o'clock போவேங்க கிரவுண்டுக்குன்னு. அவர் 5:30 டாக்டர் கிரவுண்டில் நின்றுட்டார், செக் பண்ணுறாரு, என்ன தான் பண்ணுறாரு, தூங்கிட்டு இருக்கிறாரா என்னன்னு பண்ணுறாரு. பேஷண்ட்டு வந்தார், டிவி ஸ்வீட்டில் வந்தார், டிவி ஸ்வீட்லேயே 3 round அடித்தார், 4 round அடித்தார். அதனால் நீங்கள் வந்து சொல்லணும், யூ சப்போஸ் டு சே திஸ் இஸ் ஹவு யூ ஹாவ் டு டூ இட். இல்லாட்டி பேஷண்ட்ஸ் ஆர் பேஷண்ட், lay patients, right? You cannot expect them to understand everything. Even vitamin B complex, while dispensing vitamin B complex, right? Those capsules may produce yellowish urine, right? The culprit of that particular color change is vitamin B2. What is vitamin B2? Riboflavin, right? If I am correct, riboflavin, that will produce the dark yellow color. So sometimes patient may afraid, they will not take the particular drug. So you have to such a way inform. While taking antibiotic, one of the side effect of antibiotic is diarrhea. Yes or no? So such a way, if you're, that is what communication, always I'm telling that counseling, communications are very, very important. I think you will also understand all those things. And many things I wanted to, so I'm not exceeding the time now, Dr. Suresh Kumar. Yeah. And இப்போ இருக்கிற ஸ்டூடெண்ட்ஸ்லாம் என்ன பண்ணுறாங்க, கம்ஃபர்ட் ஜோனில் இருக்கீங்க. எஸ் ஆர் நோ? லைக் ஃப்ராக் இன் எ வெல். That is what the main objective of conducting this kind of conference is, to understand each other, and you can understand what is the knowledge of the speakers from across Tamil Nadu. All are excellent speakers. Two of the speakers are my students. Many students are over here, and you can have a good network. Network is very, very important. That is the main objective of conference. Try to know, there are many take-home message. Try to take one or take-home message from this conference. And அந்த ஃப்ராக் கதையை ஒன்று சொல்கிறதான்னு சொல்கிறேன். ஃப்ராக் வந்துன்னா ஒரு தண்ணியில் ஒரு டம்ளர் ஃபுல் ஆஃப் தண்ணியில் போட்டிங்கன்னா என்ஜாய் பண்ணும். கீழே கொஞ்சம் அப்படியே கொஞ்சம் வார்ம் பண்ணிகிட்டே இருந்தீங்கன்னா, வார்ம் பண்ணால் இன்னும் கூட நல்லா ஜாலியாக இருக்கும் கிளைமேட்டுக்கு இல்லை. இன்னும் கொஞ்சம் ஹீட் பண்ணிகிட்டே, இஃப் எ கீப் ஆன் ரைசிங் தி டெம்பரேச்சர், வாட் வில் ஹேப்பன்? It has to jump out of the tumbler, right? No other way. அதுமாரி உங்களுக்கு இல்லாமல் வார்ம்லேயே வச்சுருக்குறாங்க நிறைய பேர். அதனால தான் யூ ஆர் நாட் கம்மிங் அவுட் ஆஃப் யுவர் கம்ஃபர்ட் ஜோன். அதுலேருந்து நீங்கள் யூ ஹாவ் டு கம் அவுட் ஸ்டூடெண்ட்ஸு. அதனால் மொபைல் ஃபோன் யூசேஜை கொஞ்சம் குறைச்சிங்க. I think you are all hungry with me, right? Sorry, angry with me, right? If you are telling something about the mobile phone. மொபைல் ஃபோன் யூசேஜ் வந்து நைட்டில் தான் அதிகமாக இருக்குதுன்னு கேள்விப்பட்டேன். 2 to 4 o'clock வந்து அதிகமாக இருக்குதாம்மா. ஸோ மார்னிங் என்ன பண்ணுறாங்க ஸ்டூடெண்ட்ஸ்லாம் 8 o'clock தான் ஏந்திருக்கிறாங்க. தே யூஸ் டு கெட் அப் இன் தி மார்னிங் 8 o'clock. தலையை மட்டும் சீவிட்டு காலேஜில் வந்து சாரு திட்டிடுவாருன்னு சொல்லிட்டு, எஸ் ஆர் நோ, ரைட்? So I have been the disciplined soldier throughout my life. No alcohol, no smoking, not even non-vegetarian. I think Surendra Kumar agree with me all those things, right? So I want to you be that, right? So alcohol இருக்கக்கூடாது. இந்த உங்களோட லைஃப், parents are pumping money like anything. You have to give them back. You need to shower many things to them. They should enjoy your presence. And one more thing, if your doctors makes a mistake, let me repeat, if doctors makes a mistake, a person goes 6 feet below or above the ground, below or above the ground, below the ground. A doctors make a mistake, a person goes below the 6 feet below the ground. If the lawyer makes a mistake, the person goes 6 feet above the ground. Yes or no? If your teacher make a mistake, you are all teachers. There are many teachers occupying the first 2-3 rows over here. If you make a mistake, the entire batch will wiped out. Yes or no? So what you have to do, my dear teachers? You should take care of your students, right? You just encourage our students. They are all potential enough. Only thing, they barely need of some kind of encouragement, therapy words. I expect these therapy words from the teachers and principal over here. Success people always have 2 things on their lips. கோகிலா மேடத்துக்கு பார்த்ததே எனக்கு அதான் தெரிஞ்சிச்சு. கோகிலா மேடத்தோட சக்ஸஸ் வந்து ரொம்ப சைலண்ட்டாக இருக்கிறதும் ஸ்மைலிங் ஃபேஸும் தான். இந்த ரீசன் என்னன்னா, ஸ்மைல் டு சால்வ் தி ப்ராப்ளம் அண்ட் சைலன்ஸ் டு அவாய்ட் தி ப்ராப்ளம். அதனால தான் அவங்க அமைதியாகவும் சக்ஸஸ்ஃபுல்லாகவும் இருந்துகிட்ருக்குறாங்க. And எனக்கு தெரிஞ்ச வரைக்கும் students are like birds. My dear teachers, I'm telling you, students are like birds. If you hold them tightly, they die. Yes or no? If you hold them tightly, same way if you hold them loosely, they will fly. You hold them with love and care, they will remain forever. Yes or no? Right? So I request our faculty member to take care of our students. They are really energetic and they will do it like anything. And a man asked Buddha, Lord Buddha, I want happiness. What is the question he asked? I want happiness. Immediately that Lord Buddha replied, you remove I from your sentence because I is the ego. Remove I want happiness. Remove I, then want. You remove want also, desire. Now the left is happiness. If you remove ego from you and you remove the desires, you will be always in happy mood. So I hope you will enjoy all the 3 days. You can have a good time with Shanmugha College of Pharmacy. I pray God to shower all his happiness to all the attendees over here. Thank you for the opportunity given to me. Thank you. All the best.
Thank you, sir, for sharing your short stories, which was interesting and delightful. Success is not about how high you can climb, but how you bounce back when you fall. May I now invite Dr. K.K. Sendil Kumar, Professor, Head of the Department of Pharmaceutical Analysis, Sri Shanmugha College of Pharmacy, to this 3 days National Conference on Recent Innovation in Pharmacological Research 2024 to introduce our chief guest of this inauguration ceremony.
Good morning to all. It gives immense pleasure to stand before you in this auspicious occasion. In behalf of management, I would like to invite Dr. N. Kannappan sir, M.Pharm PhD, Professor, Annamalai University, Chidambaram. I can say proudly, I was a student of Dr. N. Kannappan sir. We given some happiness. Once again, I got an opportunity to invite my staff. That really very, very happy moment. You also can enjoy and celebrate this knowledgeable session. Sir as a speaker. Thank you to accepting our invitations. And thank you for giving this opportunity to the God. May all the wishes to the universe species. For one small video clippings we made for dedicated to the Dr. N. Kannappan sir, M.Pharm PhD, Professor, Annamalai University, Chidambaram. Please, I request to play the video.
Let's have a AV on our chief guest remarkable journey.
It's with great pleasure and anticipation that we extend our warmest welcome to Dr. N. Kannappan, Professor and DRC member of Annamalai University. Dr. N. Kannappan's illustrious career and remarkable achievements in the field of pharmacy education and research stand as a testament to the dedication and expertise. With 27 years of teaching experience, Dr. N. Kannappan's contribution to academia are truly commendable. Dr. N. Kannappan's qualifications, including the successful completion of GATE 1994 and receipt of stipend at JRF during the PG program, demonstrate the commitment to academic excellence from the outset of career. Dr. N. Kannappan's research endeavors are equally impressive. Having authored 90 research papers in national and international journal and have completed 1 minor research project and 3 major research projects, Dr. N. Kannappan has significantly contributed to advancing knowledge in the field of pharmaceutical analysis. Furthermore, Dr. N. Kannappan's mentorship has had a profound impact on the next generation of scholars. Under his guidance, 38 M.Pharm students and 16 PhD scholars have been awarded, with 5 PhD scholars currently pursuing their degrees under the supervision. Dr. N. Kannappan's engagement extends beyond the classroom and laboratory, as evidenced by the participation in 33 national and international conferences, symposiums, and workshops. Dr. N. Kannappan's willingness to share his expertise through 20 special lectures in various pharmacy colleges underscores his commitment to knowledge dissemination and community engagement. His accolades, including the prestigious Pharma Recognition Award received at the Pharmacy Guru Felicitation Ceremony of Pharmacology in 2021, and the victory in the Awareness Promotion Video Competition on UN Unsustainable Development Goals for Pharmacy Teachers in Tamil Nadu, further highlights his exemplary contributions to the profession. We are honored to have Dr. N. Kannappan with us today. Your presence enriches our gathering, and we are eagerly anticipating to your insights and contributions to our shared pursuit of academic research excellence. Once again, welcome Dr. N. Kannappan. Your legacy in pharmacy education and research inspire us all.
I cordially welcome our chief guest, Dr. N. Kannappan, M.Pharm PhD, Professor and Drug Research Committee Member, Department of Pharmacy, Annamalai University, Tamil Nadu, who has a wealth of experience and knowledge that they will generously share with us today. Let us kindly invite our chief guest to the stage. Sir, please. Now, let us call our principal, Dr. P. Suresh Kumar, sir, to honor our chief guest. I kindly invite Dr. N. Kannappan to share a few words with us.
Good morning to one and all. Respected Chairman Thiru K. Shanmugam sir, respected Executive Director Mr. A. Tirumurthy, and respected Joint Secretary Tirumati Kokila Shanmugam, respected Principal Dr. Suresh Kumar, and organizing committee of this 3 days conference. Thank you for the given opportunity to deliver lecture for the session. So before that, they called me for inaugural speech, so I'll finish within 5 minutes because time is running out. Okay, so I start with Tirukural. Utravan Tirpan Marundu Ulichalvan Yana Appal Nalgutre Marundu. So 2000 years back, our Tiruvalluvar itself categorized our pharmacist profession as a Utravan is a patient, Tirpan doctor, Marundu medicine. I think you have to reply Marundu medicine, and Ulichalvan is nothing but a manufacturer of medicine and giving. So 2000 years back itself, the pharmacy is categorized by Tiruvalluvar. So it is in the, I think, 949th Kural, that is a 95th Adhikaram of Marundu. I think because I was remembering when they started playing first Tirukural. So in the first beginning of this session, they started with Tirukural. So that's why I remember that one. I want to emphasize that one. And regarding this recent students' attitude, our Dr. Arul Kumar sir is mentioned. So I want to add upon some one or two more points. That's all. So I give some information that focus. So first will be the focus. So what you are going to become in future. So you focus from the second year or third year onwards. From the first year onwards, you can focus for GPAT. Now it is a GPAT. In those days, before 2010, it was GATE. So from the first year onwards, you start preparing for GPAT. So that because in the GPAT, all the subjects are included. So start focusing from the first year onwards. So second will be self-confidence. You should have your own self-confidence. Many points have given our chief guest. And social science. You should aware about your basic social science. You should come out from the mobile phone. First thing will be mobile phone. You should come out from the mobile phone and see the world through your eye. So that is third one. And fourth one is communication. Many things our Arul Kumaran sir has mentioned about communication. So communication, connection and communication. So connection will be, you should have a national connection, you should have an international connection, you should have a global connection, the international global connection to enrich your knowledge. That is basic thing. And communication is also very important for the learning. So for the learning, the communication is very important. So only thing, since I am being the analytical person, I am having controversy with one point mentioned by our Arul Kumaran sir. Adjustment and compromise. Adjustment and compromise should not be there in pharmaceutical analysis. Or quality of drugs. So in the quality of drugs, adjustment and compromise, what will happen? The product will not be quality. So adjustment and compromise should not be there with the quality of the drugs. So in other cases, in other field, we can have adjustment and compromise. So with this few words, I thank the organizers of this function, particularly our Dr. Parimal, Dr. Gopal Satish Kumar, and other members of the committee. I thank this opportunity and I conclude my session. I conclude my inaugural address, and I will again meet you on the special lecture. Thank you, Anandal. Thank you.
Thank you, sir, for sharing your experience. Thank you, all the delegates, staff members, participants, and students who are all made this inauguration as much as the enlightened one. By this, we end our inauguration ceremony. Now I request all the delegates to take a tea break and assemble after 15 minutes for the first session in the auditorium.
It's my distinct honor and privilege to welcome our esteemed chairperson, Dr. M. Surendra Kumar, M.Pharm, PhD, Principal and Professor, Department of Pharmacology of Shanmugha College of Pharmacy. Over the years, he progressed to the role of senior lecturer and eventually assumed the position of principal, also a member of Indian Hospital Pharmacists Association and Indian Society of Pharmacology. It's with great pleasure and excitement that we gather here today to embark on this journey of knowledge, sharing, collaboration, and innovation. Let us engage in a fruitful exchange, foster new connection, and contribute to shaping a brighter future. Welcome once again. Let us make this conference an unforgettable experience for all. I kindly request Dr. M. Surendra Kumar to come up onto the stage and take a seat to listen to our esteemed speaker.
It's my distinct honor and privilege to welcome our esteemed chairperson, Dr. Mohanraj Subramaniam, currently working as a professor at the Vellalar College of Pharmacy, Tamil Nadu. He has completed his Bachelor of Pharmacy in the year 2006 and Master of Pharmacy in the branch of Pharmacology in the year of Pharmacology in the year of 2009 and its project completed in Division of Pharmacognosy and Ethnopharmacology. I kindly invite Dr. Mohanraj Subramaniam sir to come upon to the stage and have a seat. It is my distinct honor and privilege to welcome our esteemed chairperson, Dr. Rajarajan, M.Pharm PhD, for this conference. It is with great pleasure and excitement that we gathered here today to embark on this journey of knowledge sharing, collaboration, and innovation. Let us engage in fruitful exchange, foster new connections, and contribute to shaping a brighter future. Welcome once again. Let us make this conference an unforgettable experience for all. I kindly invite Dr. Rajarajan sir to come upon to a stage and have a seat.
Let me kindly invite Dr. Surendra Kumar, M.Pharm PhD, to have a few words about our esteemed speaker.
Good afternoon and all. So, speaker invite பண்றதுக்கு முன்னாடி I want to say a few words. நம்ம சீஃப் கெஸ்ட் சார் அருள்குமரன் சார் ஸ்டேஜில் பேசுகிறப்போ சார் வந்து என்னோட டீச்சருங்கிறத விட என்னோட ஸ்டார் ஸ்டூடெண்ட்னு சொல்கிறது இதாக இருக்குன்னு சொன்னாங்க. அதுக்கு முன்னாடி நான் சொல்ல விருப்பப்படுறேன். சார் ஐ எம் ஆல்வேஸ் யுவர் ஸ்டூடெண்ட். இங்கே இந்த ஸ்டேஜில் நாங்கள் நின்றுக்கிட்டு இருக்கேன் அப்படின்னா அதுக்கு முதல் காரணம் நீங்கள் தான் சார். தேங்க்யூ சார். சாருக்கு பக்கத்தில் உட்காரதே இன்னைக்கு எனக்கு கிடைச்ச ஒரு பெரிய பெருமையாக நினைக்கிறேன். ஒன்ஸ் அகேன் தேங்க்யூ சார். இட்ஸ் மை ப்ரிவிலேஜ் and proud to introduce our today's speaker, Dr. Kannappan sir, who is a professor in pharmacy with 25 years of teaching experience. He was qualified GATE in the year of 1994 and received stipend JRF during his PG program. He authored about 90 research papers in national and international journals. He also presented and participated in 33 national and international conferences, symposium, workshops, etc. Under the guidance of him, 38 M.Pharm students and 16 PhD scholars were awarded. Currently, 5 PhD scholars are pursuing PhD under him. He had completed 1 minor research project and 3 major research projects for about worth of 54 lakhs from various funding agencies. He delivered more than 20 special lectures in various pharmacy colleges. He is a member of Board of Studies in many universities. He is a reviewer of many reputed journals, including Journal of Chromatographic Sciences. He received Pharma Recognition Award in Pharmacy Guru Felicitation Ceremony of Pharmalog from the Nirali Prakash Publications, Pune, Maharashtra in 2021. He is a winner of Awareness Promotion Video Competition on UN Sustainability Development Goals for the pharmacy teachers in the state of Tamil Nadu in connection with Teachers' Day celebrations on 5th September. Sir's profile is very big, so இவ்வளோத்தையும் நான் பண்ண முடியாதுங்கிறதுனால கட் ஷார்ட் பண்ணி கொடுத்துருக்கேன். Sir, with this small introduction, I welcome you for the stage for deliver the speech.
Good afternoon to all. Once again, assuming the stage. Thank you, Dr. Surendra Kumar sir, for the nice introduction. Shall I start? கலர் மேக் இருக்கா இல்லை எப்படி தானே? Presentation. Okay. First of all, I wish to tell you what is the reason for choosing this topic. As you know very well, in the 6th semester of B.Pharm, you are studying pharmaceutical quality assurance. So in that, you will be studying about regulatory affairs. So even though there is a lack of information and there is a lack of awareness about regulatory affairs, because a lot of opportunities are available in the field of regulatory affairs. So that is the reason for choosing the first one. And then as a topic of this conference is related to pharmacology, I added bioanalytical method validation. And actually there was a previous plan was method development, but development, there is no role of regulatory affairs in the development. There is a role of regulatory affairs in the validation part only. So that's why afterwards I shifted to validation. Okay. So the next slide. The regulatory affairs plays a vital role because it starts from the research to the end. The end part will be your user, like a patient. And actually, for the development of any molecule, it takes more than 10 to 12 years of different stages, and also it involves huge money. When there is any new molecule is invented, in the beginning, it might be started with around 50 to 100 moieties or 500, even 500 moieties, and it ends up with one from the manufacturer for the marketing. So whenever the new moiety is entered into the market, there are a lot of regulatory authorities are there in the world. I think I given the details in the down. US FDA, United States of Food and Drug Administration in USA, M.H.R.A, Medicine and Healthcare Research, Regulatory Affairs in UK, and then Australia, TGA, Therapeutic Goods Administration, and then European Medical Agency, EMA. And next, so when we start with the development of any pharmaceutical product, it involves many of the following stages. So it may be from natural source, or it may be from the synthetic source, or it may be from in silico, computer drug design. So the molecule may enter into the market by using these ways, and it involves characterization by IR, NMR, mass spectra. So once you synthesize the compound related to natural source or even related to any other field of innovation, it requires the characterization by spectroscopy. And then preclinical testing, pharmacological screening and assay for therapeutic efficacy, preclinical testing, clinical studies including regulatory innovations, regulatory approvals for the commercialization of new chemical entity and new biological entity. So these are all the different stages of development of new drug. In the development of new drug, the drug approval process is a long process. It requires all the information about from the starting point to the end point of the product, even from the preclinical studies to the post-marketing surveillance studies. So there are many clinical studies you have studied in the pharmacology, clinical trials. Next. It starts from risk discovery, preclinical testing, clinical trials, phase 1, phase 2, phase 3, product sale, and then phase 4 trial. Phase 4 is post-marketing surveillance, and these are different stages. I think M.Pharm pharmacology people might be knowing very well. Even pharmacology, in the pharmacology also you might be studied. Next. And in preclinical and clinical trials, so after the synthesis, the preclinical study starts, and they will file the IND. After the preclinical studies is completed, then they will find for the investigational new drug. So all these things you might be studied in the quality assurance. IND is a process starting before investigational new drug. So without applying for investigational IND application, no one should start any research related to product for the market. For the project work of the B Pharm or M Pharm, you can do the research work without IND because that is not for commercialization. If you want to go for the commercialization, definitely you should go for the application of IND. And then in the FDA will review the data. So what are the data you have to submit to the FDA, Food and Drug Administration? That is your preclinical data, chemistry, manufacturing details, dosage form, investigational protocol. So details of investigator, concern of participants for the clinical trial, and other agreements. And these phase 1, phase 2, phase 3 and phase 4 are the stages of various clinical trials, as you know very well. And preclinical evaluation involves demonstrating the pharmacological effect of lead molecules using suitable animal model. And disease-oriented or organ-oriented further will be developed that toxicological studies, pharmacodynamic studies and pharmacokinetic studies, as you know very well. I don't want to explain all those things because this is a basic thing. So next one. And these are all the different approximate phases of year. So for the first drug discovery and preclinical trials, it requires minimum 4 years. And then 2 years for phase 1 clinical trial and approximately 4 years for phase 2 clinical trial. And then another 2 years for phase 3 clinical trial. After completing the phase 3 clinical trial, we can go for the NDA, new drug application. Once the NDA is approved by the FDA, then we can go for the marketing. So once the NDA is approved by the FDA, we can go for the marketing. After marketing, the phase 4 will be started. So we need not bother about the phase 4. Phase 4 is a automatic process after the marketing. Next. Phase 1, phase 2, what are the different establishment of phase 1, phase 2, phase 3? The phase 1 is a healthy human volunteer, a small group, it will be 2200 volunteers, 6 to 9 months to establish the determination of pharmacokinetic difference between animals and human. And then phase 2 will be diseased population, so natural or induced disease population, and the number of volunteers, and 6 months to 3 years to determine the effectiveness and safety of the drugs. It will be established in the phase 2. And then the later phase of the phase 2, early phase and later phase, 2 things will be there. That is a number of participants will be differ, and also the single blind study and double blind study. I think you might be knowing single blind study and double blind study. So to avoid the bias, so single blind study and double blind study is used to avoid the bias. So single blind study means the innovator may know the name of the drug and the operator will know the name of the drugs, but only the patient or volunteer. So volunteer does not know the name of the drug, so that it will be unbiased. And the double blind study will be both the innovator will be knowing, but only the operator, means your operator and patient, there is your volunteer may not know the name of the drug and for what purpose they are taking, so that to avoid expecting the result. So to avoid expecting the results, who is receiving placebo and who is receiving the drug. So they'll give the code numbers and they'll give the sample codes, no names. And the phase 3 will be 200 to 1000 plus volunteers, 1 to 4 years. And to establish the safety and efficacy of the drug candidates. And next one. And this is a diagrammatic representation of the phase 1, phase 2, phase 3. In phase 1, pharmacokinetic profile will be established. Pharmacokinetic absorption, distribution, metabolism and excretion. And then determination of safe and tolerable dose. What is a safe dose can also be established in phase 1. And then predictable toxicity from the phase 1. And phase 2, safety and effectiveness of the test compound. Recording of short-term side effects in health impaired patients. And then determination of the dose range will also be established in phase 2. And in phase 3, recording of labeling information. So labeling information includes what is the type of packaging, what is the type of formulation, what is the time duration of expiry date and storage conditions. All those things will be established in phase 3. Depends on the stability of the product. And recording of risk benefit ratio. Usually risk benefit ratio should be more, risk ratio should be less, risk benefit ratio. And then recording of less common side effect. Less common side effect will be observed in the very few volunteers. it may not be observed in all the volunteers. It will be observed in the very few volunteers. So that also will be recorded. Everything should be recorded. So regulatory affairs need all the data in documentation with record. So they need all the documentation. So documentation part is the most vital part in the regulatory affairs. So next. And in phase 3, several correspondence between, actually when there is any doubt between the regulatory authority and sponsor, they will get the communication between them. If any clarification, they will be asking you every time, and then they will be clarified. And if FDA is satisfied with all the details which is submitted by us, then they will give the new drug status under controlled marketing, NDA, new drug application. So we are going to see each and every part in a small way. So DMF, IND, NDA and ANDA. So phase 3, the second part of the phase 3 will be starts before approval of the additional safety data. So it includes cost and effectiveness of the drug against other available drugs in the market. Comparative study between our drug and the same drug, same type of drugs available in the market. Next. And phase 4, as I mentioned earlier, phase 4 is a market surveillance, post-marketing surveillance. So phase 4 will be, this is also controlled looking like controlled marketing, so that we can extend our product to the little more number of patients or little more number of participants. So that it will be 5,000, 10,000. So we have to catch some physicians patient, and we have to monitor. So it should not be free marketing. It should be a controlled or restricted marketing, so that we can find out what are the rare side effects. We can find out rare side effects which is observed in the post-marketing surveillance. So the group should be within our control. At any time we can withdraw the product from the market. Like that, it will be in it should be in our hand. So manufacturers should gather the report and such new information to respective regulatory authorities, because we have to communicate to the regulatory authorities regarding the post-marketing surveillance report. So that is very important. That is also will be monitored by the regulatory authorities. Next. And these are the drug development process, starting from molecular biology, biochemistry, medicinal chemistry, pharmacology, toxicology, because every field is in our every field is interrelated. Without one discipline, we cannot do anything. Because nowadays, drug regulatory division also a part of a drug development process. Next. And functions of regulatory affairs department to keep track on the ever-changing legislation, because every day the rules and regulations are changing. So we should be aware about the rules and regulations, which is time to time, which is updated by the regulatory authorities. And need to study the process of drug development. Data generated during preclinical and clinical data should be reported, and QC procedures should be aware, and then safety and efficacy of the product will be maintained. Next. And the regulatory affairs personnel should give a strategic and technical advice to the concerned departments, for example, R&D, manufacturing, marketing, relations, and to the particular, to the product rules and regulations. Because they are the bridge between all the departments. So they should give the information about all the product, because they have data from the different departments, and they only will be filed. So they will be collecting the data from the pharmacology department. They will be collecting the data from the toxicology department. They will collect the data from the quality control department, quality assurance department, all those things, and then they will be compiling the data and will be submitted to the FDR regulatory authorities of the particular respective countries. And the regulatory affairs personnel requires communicative competence, both written and oral, analytical abilities, presentation skills, understanding of both legal and scientific matters. So this is a basic requirements of the regulatory affairs personnel. So if you want to become a regulatory affairs personnel, you should have these abilities to make a good documentation, to make a good presentation, to make a good analytical abilities, all those things. So because a lot of opportunities are available in the regulatory affairs, because it is unaware by the people in the field of pharmacy. So next, and we'll see one by one with few slides about the DMF, IND, ND and ANDA. Already I have studied in subjects. So next. So drug master file is a confidential document. It is there. It may be a facility, process or article. It is used in the manufacturing, processing, packaging and storaging. And actually it is a confidential documentation to the FDA. If any, actually there is a misunderstanding about DMF by the people. So DMF means drug master file. So drug master file means whenever I ask questions to the student during Viva, what is DMF? They used to say drug master file. And what is drug master file means? It is information about the drug. It is information about the drug only. But for what purpose it is used? What is the purpose of drug master file? Anybody? From third year, fourth year? Fourth year I think might be completed the third year quality assurance, sixth semester. So actually drug master file is it will be filed to the FDA and FDA will review the data and will keep in the shelf and they will give the data to the people, those who are buying the product from the particular manufacturer. So it is a confidential document. It is not an open document. It is a confidential document. And once the consent is given from the manufacturer or innovator, then they can disclose. So the drug master file is a confidential document. It will not be disclosed to all the people. It will be filed to the FDA. FDA will review. Without review, they will not keep it. FDA will review. And after the review, they will keep it. And then they will be revealing to the people, those who need to buy the product from the particular manufacturer. So that is the drug master file. I think all the informations are given. Next one. There are different types of drug master file. Manufacturing site. We can document manufacturing site, facilities, operating procedures, and personnel also we can make it. And then type 2 will be drug substance intermediate. If you have any procedure for the intermediate preparation, then you can submit a drug master file in particular intermediate. So if anybody wants to buy that particular intermediate, they can go to the DMF with your consent letter and they will ask the quality of that particular material. So they will reveal the thing so that if you satisfy, then you can buy or you can purchase from the. So your FDA is a third party. FDA is like a justice, like a judge. So they will give you the information about the product. So next, investigation new drug application, IND. Once if you want to start any new investigation, you should get the approval from the FDA before going for the investigation for commercialization, for commercial purpose. So not for all the thing. Investigation drug, investigation new drug, it may be investigation medicinal product or it may be next. Next one. Okay, so the investigator IND. So there are different types of IND. So investigator IND submitted by the physician who both initiates and conducts the investigation and under whose immediate directions the investigation new drug. So this thing, if you read the thing, you will be boring. If I give the example and if I give any additional points, then only you can able to understand. So the new indication for old drug. So new indication, if there is any new indication for old drug, that is called, for that also we should apply for IND. So for example, aspirin. In those days when we studied, aspirin is used for in 90s, 80s, because it is a very old drug, 60s, 70s. It was used for aspirin, analgesic. Now it is used for antilipidemic agent. I think so. I think 60 milligrams, 30 milligrams is available in the market. So that is a new indication. I think from 2000 onwards or 1990 onwards, that is a new indication because old indication is, old use is analgesic, but nowadays nobody is using aspirin for analgesic. Nowadays it is used for the anti-atherosclerosis drug. So like that, for old drug, any new indication, definitely we should go for the IND. So without IND, we should not reveal any information about the old drug. Next, emergency IND. So emergency IND, no need to give any information or no need to give any example. Very best example is our corona vaccine. So if there is any epidemic or if there is any endemic or if there is any pandemic in the world, definitely there will be a emergency IND will be given. So with the basic data, they will give the approval. Once after completing the details, then we can go for the marketing. Next, treatment IND. If any particular treatment, if you are going for any IND, you can apply for the treatment IND also. So next, any serious conditions are like that. So this is a flow diagram of IND. Starts from your applicant IND that will be reviewed by CDER, Center for Drug Evaluation Research. CDER is actually I am focusing on the US FDA. So this is from the US FDA. So here under US FDA, the CDER is one of the department. I mean, it's a very big department, not like your simple department. It will have all the facilities to test all the materials. So they have, they will be checking medical, clinical facility, chemistry, pharmacological toxicology, and statistical review. They will do all the review. That is reviewed by the CDR. Once if they satisfy, then it will be going for the safety review, safety of our product. Then safety acceptable means then it will be complete reviews, and then review complete means acceptable. No deficiency means then it will be study ongoing. That is a green signal. So last one is up to green signal, it should pass the path. If there is any deficiency in between, then it will be reviewed by the sponsor. It will be given back to the sponsor. They will ask the sponsor to resubmit within the stipulated period of time, around 2 months or 3 months or whatever maybe. So next will be new drug application, NDA. The new drug application is a critical component in the drug approval process, because once the IND is finished, once IND is approved, then we can go for the marketing. For marketing, we should apply for the new drug application. Once the new drug application process is over, then you can go for the marketing. During the marketing, while submitting for the marketing, it will be under the restricted marketing. So next one, this is a review of or US FDA for the different types of process. Biopharmaceutical, pharmacology, statistics, microbiology, chemistry, medical. So all the things will be reviewed by whatever the data we are submitting to the FDA. They will be reviewing all the data. They will not just like that. They will not approve. They will be reviewing each and every line by performing the experiment, by performing, not by reading, by performing. So you have to supply the sample. We have to supply our sample. And they will take minimum 3 months to 6 months. Sometime it may take 1 year also. So this is the hurdles period for the drug approval process. So these are all the things. So it may take 6 months or it may take even 1 year also for the review. After that, they will be revert back to you for the review. That they will give you any deficiency points. They will be telling you. Then we have to rectify all the deficiency points. Then we have to resubmit the data for the rectification. Then they will be reviewing. So it is a continuous cycle process. So till the product is they feel it is a quality, till that it will be review. So next, about ANDA. So because NDA will be given for the particular manufacturer or particular innovator only. So the NDA period will be our patent period. So the patent will be provided, means that is called your proprietary preparation. So they will be awarded a patent to manufacture only the particular manufacturer. And once the patent period is over, around minimum 15 to 20 years, then the product will be open for market. Once the NDA is, once after patent period is over, then everyone can enter into the market to market the product. So that is called your generic. Generic are different trade materials or brand names, different brand names. So what is the difference between the innovator product and generic product, or trade product, trademark and generic? I think you might be aware about what is the difference between trademark and generic. So trademark will be Any response from the audience? What is the difference between trademark and generic? In generic, I think nowadays, Jan Aushadhi medical shops, all the generic drugs are available. Makkal Marandagam, generic drugs are available. So what is the difference you observe? Generic products are cheaper. Generic products are cheaper means not in the quality, by the cost, less cost. And brand name will be more cost because they spent 5,000 crores money, so that they have to get back the money from the patient. So they fix the price on their own till the patent period is over. So once the patent period is over, then everyone can enter into the market. The price will be reduced. So even after patent period is over, everyone can enter into the market as a brand name also. So different manufacturer with a different brand name. So different manufacturer with a different brand name will also reduce the cost. It will come down to the 10-fold reduced cost will be there. Sometime in the beginning it may be 150 rupees. After the after the patent period is over, once NDA is opened, ANDA is open, abbreviated new drug application is open, then it will be come down to the 15 rupees. So that is a benefit for the patient. And once if you go for the generic, then it will come down, even it may be available for 5 rupees or 6 rupees. And then orange book is available, which is the approved drugs are given in that orange book. Next one. Okay, orange book, it's pharmaceutical equivalent. So actually, the product which should be pharmaceutically equivalent, it should be pharmaceutically equivalent, bioequivalence, bioavailable, similar bioavailability than that of the marketed product. Because if you want to manufacture the same product in different way, that's why, that's all. So you should not change any indications. Any indication should not change. For example, if you take any drug, for example, recently in 2019, some anti-diabetic drugs patent was over. So once the patent was over, next day itself, 10 manufacturers came forward for the different trade names, so that they will manufacture the same drug with a different brand name. That's all. So that it should be bioequivalent. It should be bioequivalent that of already existing in the market. It should be similar bioavailability that of existing market. So and also mostly pharmaceutically also should be equivalent. For example, strength may be at 20 milligram means your drug strength also should be 20 milligram. So like that there is a thing. Next. And this is your again same flow diagram of your ANDA, and then again the bioequivalence review, chemistry and microbiological review, request for the plant inspection. They need whether you have a facilities or not. The USFDA people may come and visit your manufacturing unit and find out the facilities. And then labeling review. So labeling review is very important for the ANDA, because we should be equivalent to the product which is available in the market, already available in the market. So that strength, storage condition, all those things will be similar. Mostly it will be self-explanatory. Next one. Next slide. Okay. So we'll see some different organization of our CDSO in India. So regulatory authorities in India will be CDSO. You might be studied about very well. What is CDSO? Is there? So Central Drug Standard Control Organization. So in India, we are following CDSO approval process. And we have a different, DCGI is head of the institution, and one headquarter, and then 6 zonal offices. Sub-zonal offices are 5. Actually, zonal offices includes Chennai, Bangalore, Chennai, Calcutta, Mumbai, Delhi, and other places. And the sub-zonal offices, port and airports are also available. So wherever there is a export and import, to govern the export and import, ports and airports are governing. 13 ports and airports are there. and then 6 laboratories. There's a CDL, Central Drug Laboratory, Central Drug Testing Laboratory, and Regional Drug Testing Laboratories, and IP Commission. So these are all the organizations, these are all the activities followed by CDSCO, means activities of CDSCO. So it will be monitoring all the drug testing laboratories. So now they are going to start, I think next part is there, medical devices testing laboratories. So next, these are the major schedules which comes under the regulation. Schedule M, M1, M2, M3, T and UN. Actually other schedules you might be know very well, M1, M2. M3 is a new schedule. Schedule M3 is a new schedule which is for medical devices. Even though it becomes old, because 2017 they started this M3, and from 1st January 2018 onwards it is implementing. Now the even Drugs and Cosmetics Act is going to revised as a drugs, medical devices and the cosmetics act. The later part of the PowerPoint is giving the information. So next, major responsibilities of the CDSCO is conduct training program, approve the licensing of manufacturing certain categories of drugs, regulate the clinical regulations in India, regulate the standards of imported drugs, and publication of IP. Okay. Next, and regulatory control over the import of drugs, and these are the continuing our duties of our CDSCO. Amendment of Drugs and Cosmetics Act and rules, license for approval of blood banks, large volume parenterals, vaccines, rDNA products, and some medical devices. So banning of drugs and cosmetics. So they will be releasing every 3 months once, 4 months once. They will be releasing the list of drugs. You can visit the CDSCO website to find out all the circulars and other notices. Grant of test licenses, personal licenses, and NOCs for the export and testing of new drugs. So next. State government licensing authorities, licensing of drug testing laboratories, approval of drug formulations and for manufacturing, monitoring of quality of drugs, recall of substandard drugs, and investigation and prosecution in respect to illegal provisions. Actually, a major role of their regulatory authorities will be to implement or to focus on the quality of the product. So they will be visiting all the manufacturing sites and even retail sites, and they will be taking 8 samples per month. Each and every drug inspector will take 8 samples per, I think you might be knowing, 8 samples per month, 5 samples from the private sector and 3 samples from the government sector. And they will be, because it's a periodical checking, random checking. So randomly they will be selecting any 5 brands, any 5 products from the market, and any 3 products from the government hospitals and government organization. And they'll be keep on sending the product to the drug testing laboratories to find out by this way only they found out many substandard drugs and unqualified drugs. So next, and this is one of the notice which is given in 2022 July to revise the Drugs and Cosmetics Act, but still it is not revised. Still 2024 comes, still it is not revised. It is still under the process of revising. I'll give you some I'll give the information about what are the different criteria are covered in the new bill. Next. So the major provisions of the new bill 2023, even 24 also came, not regulated. So regulation, it seeks to regulate the e-pharmacies, because nowadays everywhere you see the e-pharmacies. So e-pharmacies is a anti-pharmacist activity. So without pharmacist, they are selling drugs like Zaros or for food delivery, they are doing. So that is regulated by the new bill. That is regulated by the new bill, but still it is not implemented. Devices and provides for penalties include imprisonment for failing to pay the compensation for injury or death during clinical trial for both drugs and medical devices. So this is a new amendment because as I mentioned earlier, we should be aware about the latest regulation amendments. We should aware about all the regulatory amendments. And Drugs and Cosmetics Act 1940. So previously in the 1940, it was started as a Drugs Act. Not even cosmetics was included in 1940. It was called as a Drugs Act. So Drugs Act 1940. Then in 1960s, they included cosmetics. So Drugs and Cosmetics Act 1940 only, but there is a little amendment in the name cosmetics. Cosmetics also included as a product. And then now 2022 they are going to implement Drugs, Medical Devices and Cosmetics Bill. Bill means before approval. Before approval. After the approval it will be called as Act. Next, to replace the old one. Okay. Actually in the Drugs, Medical Device and Cosmetics Bill 2023 or 24, 25, when it comes we don't know. And the draft bill has the separate chapter for the Ayush. Previously there was not a separate chapter for Ayush. Ayush includes SOVA RIPR. So SOVA RIPR is a East Asian countries traditional medicine like Tibet, China, all those things. Tibet, Bangla means this East Asian country. So East Asian countries traditional medicine is called like our Siddha. In South Indian medicine is Siddha. Ayurveda is a Indian system of medicine. So like this SOVA RIPR and homeopathy also included in the thing. So new definitions, draft bill introduces various new definitions or provisions like bioequivalent studies, bioavailability studies, clinical trial, clinical investigation, and even controlling authority for the OTC product, over the counter product, all those things. Next. And penalties also will be revised. Compensation also is given. So penalties also, because penalties 1960s, they fixed the penalty in 1960s around like 1,000 rupees, 2,000 rupees, sometimes 5,000 rupees. Now 1,000 rupees, 2,000 rupees is nothing. So they are going to revise the penalties. And then even e-pharmacy is going to revamp. Next. Medical devices testing centers. So the recently our finance minister announced that in the interim budget they are going to start the many laboratories which is required for the testing of medical devices, and also they are going to start provisions necessary fund for establishing the area of medical devices testing centers and approval to include a new PG course. It may be in future there may be a M.Pharm medical devices, because now there is available M.Pharm regulatory affairs, and many of universities in Tamil Nadu and India also they are pursuing M.Pharm regulatory affairs. In future there may be a M.Pharm medical devices. Next. So second part of the lecture will be bioanalytical technique validation, means bioanalytical method validation. And as per the since this topic will be related to your topic of conference, in this actually why I am chosen this one, nowadays many people are doing research in the bioanalytical technique like LCMS, all those things. So many people are having a misunderstanding about the bioanalytical field, because normal analytical field is different from bioanalytical field. In normal analytical field will be, normal analytical development will be based on the matrix from the tablets, capsules, all the pharmaceutical formulations. But bioanalytical field involves or includes your biological samples. What are the biological samples? I think it is given. Biological matrix. So biological matrix includes blood, CSF, cerebrospinal fluid, urine, sweat, lacrimal fluid, all those things will be biological fluid, because biological fluid is different from the, biological matrix is different from the your pharmaceutical matrix. So pharmaceutical matrix does not have any interference. We can avoid the pharmaceutical matrix by avoiding some extraction procedure or any other procedure we can avoid. Even by HPLC also we can avoid the separation technique. But in the biological matrix, most of the biological fluids are having a more contents, more chemical components, so that we should have a separate entity. So that's why the two guidelines are given, M10, that's a miscellaneous guideline of ICH guideline, Bioanalytical Method Validation and Study Sample Analysis 2022, and US FDA CDR guidelines and Bioanalytical Method Validation 2018. Actually, next one, that is a definition of that bioanalytical validation is given in the down. Estimation of drugs and metabolites in biological fluids, blood, CSF, urine, sweat and lacrimal fluids. So characteristic analytical procedures and their validation in an integral part of the drug discovery process. Because in the drug discovery process, the analytical part is a major important part, because analytical part only will find out the quantity of the drug which is present in the formulation, quantity of the drug which is present in the biological fluid. So without the good analytical technique, you cannot come to conclusion of your report. So the bioequivalent study and bioavailability studies are important to find out after patent period is over to get the approval under ANDA. So I already mentioned this one. Next. And this is the different stages of your bioanalytical method validation. First sample will be there, and then sample will be processed according to the nature of the sample. And then we have to choose any one analytical technique. So choosing analytical technique, after choosing the analytical technique, then we have to go for the method development and optimization. So after choosing the analytical method, we have to go for the procedure. We have to start with which procedure we are going to follow. So the method development and optimization. So in the up to this, the top part, sample processing, analytical technique, and method development and optimization, the regulatory part will not come. The top part, regulatory authorities will not come into this picture. But in this down part, when you see the method validation and documentation, application of method to the study pharmacokinetic parameters, in these two, that's why I given in a little big, and also the content also good big. So these two area will be governed by regulatory authorities. So next one, regulatory requirements for bioanalytical method validation. So for analytical method development, there is no need to follow any regulatory requirements, as I mentioned earlier. So for the development of analytical method, we need not get any permission from any authority. We can start our own method. We can have our own method. We can have our own analytical technique. We can have our own instrument, whatever the thing. And but the method validation, it is necessary to follow the regulatory requirements, because it is a code of conduct. We have to follow with yardstick, without deviation of even a single line. And after collecting the blood, it will be processed to get the plasma, because bioanalytical method involves 90% of the sample will be in the blood sample. So after the blood sample collection, directly the blood sample cannot be injected into the HPLC or LCMS or any other GC or whatever it may be. So it will be processed to get the plasma. So plasma, with coagulants, and without coagulants, if you centrifuge without coagulants, will be serum. After centrifuging, the supernatant liquid will be collected. That will be injected into the blood. LCMS or whatever. So directly plasma or serum cannot be injected into the HPLC, and need additional procedure to avoid the blood constituents. So there are three methods: protein precipitation. So the protein precipitation means because a blood product contains protein. What are the proteins? What are the proteins? Even first year students can also answer. Albumin, globulin, fibrinogen, and all those things are there with RBC, WBC, platelets, all those things are there. So to avoid those things, they are using the protein precipitation method. And then solid phase extraction. So column chromatography is used for the solid phase extraction to avoid a particular thing. And then liquid extraction. If the sample is liquid, then we can go for the liquid. According to the polarity of the sample, we can go for the liquid extraction. Actually, these things requires many skills to adapt the liquid extraction, because we need actually I am focusing only the regulation part, not the development part. So only the regulation part. Next, sensitivity. Actually, from here onwards the regulation starts. So sensitivity, the instrument are more sensitive to detect the analyte in picogram level. So the many drugs are available in the market with the milligram level. We have to convert the milligram level into microgram level, nanogram level, and picogram level by dilution, by various dilution. So why we are going for dilution? Because when you go to the laboratory, they will ask you to, the procedure will give take 100 milligram of drug and dilute to 100 ml with any solvent, and then further make 10 ml to 100 ml, 10 ml to 100 ml. So we'll be having procedure. So what is the reason? Because the instrument is more sensitive to find out the less concentration. So to bring down to the concentration, we are diluting. And the sample preparation is based on the sensitivity of the instrument. That's why we are making dilution. So reference standards, it should have a high purity. Reference standard will be supplied by the pharmacopoeial standards. So the pharmacopoeial reference, as I mentioned earlier, that CDL, Central Drug Laboratory, many government laboratories will provide the reference standard. So reference standard is costlier and also pure and will be having a required standard. So it will be according to the BPRS or IPRS, whatever maybe IPA reference standard or BP reference standard or USP reference standard. And then internal standard will be, working standard will be little, because working standard will be little less costly, but that is also supplied by the government organizations. Internal standard will be, it is necessary to use the IS in all the samples analysis because to compare your drug. So that later part we will be seeing, because in many slides IS is there. It may be a stable isotope or it may be a structural related compound. And why IS? So to check the variability of the drug with the, because if there is no two drugs are there, then we have to go for any one IS with the name of that particular drug. So calibration curve standard to be established with this 6 to 8. So here only there is a confusion between the researchers how to fix the concentration, how to fix the 6 to 8 concentration for the linearity or for the construction of the calibration curve. So we cannot fix the linearity or construction of the calibration curve on our own. We have to fix according to its guideline. So that guideline is given. So once if you follow the guideline, you can able to understand, because whenever we are asking question to the researchers, they are not able to answer why they choose the particular concentration for the construction of calibration curve. They simply telling that my guide is instructed me to prepare this solution, so that I prepared. So there is a reason. Okay, so next one. 6 to 8 concentrations with non-zero concentration. There should not be any zero concentration. The line should not cross through 0. It is not necessary to pass through the origin 0. Okay, and a blank without analyte and 0 with IS only. 0 means IS. We can have an internal standard. When we have an internal standard, our drug will be absent. Since our drug will be absent, then it will be 0 only. Drug will be 0. And that is called with plasma serum, and that is also with how to fix the range for the calibration curve as per the USFDA guideline. So there is a LLOQ and ULOQ. So the LLOQ is a lower limit of quantitation and ULOQ is a upper limit of quantification. So lower limit of quantitation, upper limit of quantitation. LOQ, LOD might be studied in the normal validation. So normal analytical method validation, Q to R1, Q to R2, and then now Q to R14 is also going to come, Q to 14, and that is your LOQ and LOD. So it is related to LOD and LOQ, limit of detection, limit of quantitation. So limit of lower limit of quantitation and upper limit of. So a lower limit of quantitation should be at least 5% of Cmax of less or less. So we must find out Cmax. So what is the concentration maximum of drug in the blood? So without knowing the Cmax, we cannot fix the CC. So CC is you are following? Calibration curve, very good. So calibration curve. So we should not follow the calibration. We should use the Cmax for calculating the CC, for constructing the CC. And then, okay. So the ULOQ will be, ULOQ is the final one, 8th one. So LLOQ is the first concentration and ULOQ is the 8th concentration, 8th one, last one. So middle, we can have a little hierarchy. So next, if previous one, if Cmax is 50 nanogram, then it may be 100 milligram, 100 nanogram. So you can understand this one. If Cmax is 50 nanogram in the LLOQ, it may be 1 nanogram means 5% of 50 nanogram is 2.5 nanogram. 50 nanogram la 5% is 2.5. That telling it is that maybe it may be a 50 nanogram or less. I think one word is missing. Less than 50, less than it is there. Cmax or less. So 5% of the Cmax are less. So less means it should be less than 2. It should be less than 2. So less than 2 means definitely you can have a 1 milligram. That's why LLOQ will be 1 nanogram, sorry, 1 nanogram. And then ULOQ will be 1.5 to 2 times more. So 1.5 to 2 times more means definitely 50 nanogram to be 100 nanogram. I think not necessary to explain that one. Next. And the Cmax is a maximum Cmax of the particular drug, and then lower limit of standard, lower limit means lower limit of quantitation, upper limit of quantitation will be, first one is the standard 1, standard 8. Next one. Apart from this, quality control standard is another one. So quality control standard also should be established for the each and every thing. So QC standard is different from CC standard. So in the quality control standard, LLOQ QC, that's called lower limit of quantitation hyphen QC. Lower limit of quantitation QC, that will be half of the LLOQ. What I have seen earlier, LLOQ is 1 nanogram. So half of the LLOQ is 0.5 nanogram. So just for your understanding, I am giving, not for all the drugs. For different drugs, the different Cmax will be there. So just for your understanding, one number I am giving you. So next will be LQC. So that's called lower quality control sample, middle quality control sample, medium quality control sample, higher quality control sample. 3 samples are there. So lower, medium, and higher. And the lower quality control sample will be 3 times of LLOQ. LLOQ is 1 nanogram. So 3 times of LLOQ means 3 nanogram. And then middle means medium quality control sample will be 30 to 50% of LLOQ. So ULOQ. So ULOQ is a 100 nanogram. 50% of ULOQ will be 50 nanogram. So next one is a higher quality control sample. That will be more than 75% of ULOQ. So ULOQ is 50 nanogram in our case. Sorry, ULOQ is 100 nanogram in our case. And thing will be more than 75 now, we can have 85. So these 3 sample also should be included in the 8 cc. This is also included in the 8 cc of 8. Next one. Okay, so in this you can understand standard 1, standard 2, standard 3 up to standard 8. Okay, so in this LLOQ is 1 nanogram and this is double. Standard 2 will be double, standard 3 will be again double, and then standard 4 we can have, actually when we come from the last, it will be 8th one will be the 100 nanogram, that is a ULOQ, and from that HQC will be 85 nanogram, that is also in between your thing. So next will be your 30% difference will be 70 nanogram, the next will be 50% difference will be 30 nanogram, then will be around 10 nanogram difference of this. So this is a way of constructing a calibration curve by using the guideline. So by using this guideline, they are fixing your concentration for the various various standards. Next. At least 67% of the QC should be plus or minus 15% of the nominal value. More than 50% of the quality control test quality controls each level should be plus or minus 15% of nominal value. Actually, when we see the normal limit of validation parameter, it will be only 2% or 5, maximum at the maximum 2 to 5%. But here they have given a 15% deviation. So deviation is for the why? What is the reason? Any idea? Why they have given a more number of deviation, more percentage deviation? Because the biological fluids are uncertain. Because biological fluids are very uncertain. Sometime may be more, sometime may be less. It will be including, it will be interfering. So all those things. So that's why they have given a limit for a little more. Even sometime 25% deviation also is there in some cases. Case by case it will be differs. And then bioanalytical method validation parameters will be selectivity, specificity, carryover, precision and accuracy, robustness and ruggedness, matrix effect, recovery. These are all the parameters given in the regulation. As per our analytical method validation, like accuracy, precision, linearity, recovery, range, limit of detection, limit of quantitation, robustness, rigorousness, and system solubility parameter in the normal validation parameter. But these are all the parameters. Because new things will be here, carryover, and then matrix effect, dilution integrity, reinjection reproducibility. These things are new thing when we compare with normal analytical method validation. So next. So selectively detect the analyte on interest, because what is selectivity? So selectivity mostly you may know. So selectively it should, because we have to develop a method which selectively estimate the particular drug, not other drug. For example, if the formulation containing 2 drugs means, if you want to estimate the particular drug, then we have to select that, we have to find out the selectively, we have to, it should determine the particular drug. And the 6 different plot of plasma, the one hemolyzed and one lipolyzed, because 2 extreme levels. Prepare blank and LLOQ 1 nanogram for each plasma. So acceptance criteria will be, area in all blank samples will be at least RT, retention time should be 20% less than that of the area of LLOQ. So LLOQ 20 times less than that of the RT of the analyte should be less. And RT of the IS should be less than 5% of the A of the LLOQ. So this is a acceptance criteria. So next will be specificity. So for the specificity also, specifically it should determine the drug. Whatever the method you develop, specifically it should, there may be the possibility of presence of some impurity, there may be the possibility of presence of some matrix, means or in tablets it will be excipients, in capsule it will be excipients. In pharmaceutical formulation, excipient is a matrix. So combined thing. But in in bioanalytical field, it will be fluids, means body fluids or biological fluids. So matrix effect also should be avoided. And then co-medicament present. So presence of co-medicament also will be avoided. So these are all the specificity. And the limit also will be same as that of the previous one. So next one, carryover. So carryover is nothing but just like washing. So carryover means because we are using the very sensitive instruments. So instrument, even the sample holder may keep the old drug, so that it should be inappropriate washing. So the word will be inappropriate. So same word, it is a typographical error. So the reason will be high concentration of analyte will absorb onto the side of the any container, sample container, so that it will give the wrong result. So that we have to wash properly before going for the next analysis, even before going for the next concentration. Same drug with a different concentration. I think you might be studied in spectrophlorimetry. In spectrophlorimetry, fluorescence spectroscopy, we should start with the lower concentration to the higher concentration. So because lower concentration will not be affect the higher concentration. For example, if you are preparing 1 to 5 microgram per ml, we have to scan first 1 microgram, then 2, then 3, then 4, then 5. We should not come from 5, 4, 3, 2, 1. If you come from 5, then 5 microgram per ml solution will influence with the 4 microgram per ml. So that there is a absorption in spectrophlorimetry, absorption effect will be possible. Here will be a thing. Remedy will be extend the duration of washing. So changing mobile phase and diluent. Next, accuracy and precision. I think a normal thing. Accuracy is close to the true value, and precision will be when we repeat it, you should get the same value. I think definition is given. I think time running out of time. So next slide. This measurement are precise but not accurate. There is a relationship between accuracy and precision. So our target is center point, and if you get the result in the other side, all the results are in the same area. That is called your. This is not accurate, but it is precise. This sample is precise because, for example, your target is 100%. Your target is 100%, but you are getting the result 69, 70, 71, 69.5, 70.5. All will be around 70. So all the values are precise. Precise means the values which is close to each other. All the values are close to each other. It is precise but not accurate. Okay, next. And these are all the other examples. The measurement is accurate but not precise. Center point is the accuracy is there, but precise, not precise. And the next one is about the measurement is neither accurate, not precise. Not accurate and also not precise. Next one. Measurements are precise and accurate. So this is a just a diagrammatic representation of the measurement of precision. Next one. Robustness ruggedness you may be knowing. So deliberate deviation, the method parameter, same as that of your normal analytical method validation. And the ruggedness will be analysis by different instrument, different person, different people, different laboratory. So next one. Direct or indirect alterations of matrix effect. So matrix effect means due to the presence of excipient, due to the presence of molecules, due to the presence of other biological fluids. And to avoid more selective extraction procedure will be used, changing buffer and extracting solvents, use of stable internal standard. So next one. And recovery study, it's 100, similar to that of your analytical method validation. Only the thing will be percentage deviation is a little more extended. Percentage deviation is a little more extended. And acceptance criteria will be percentage of CV. Actually, we have to calculate here percentage RSD. Percentage RSD and percentage CV is more or less same. Coefficient of variation and relative standard deviation, percentage RSD, relative, because we have to convert the data into the standard deviation. After converting data into the standard deviation, we have to convert into relative standard deviation. So relative standard deviation will be giving you the clear picture about the percentage deviation. So next. Dilution integrity and reinjection reproducibility. So dilution integrity, because once if you want to prepare a CC calibration curve, if your sample is beyond the calibration curve, then we have to bring down to the lower concentration. So for the dilution integrity, so we need to develop a skill to find out to bring the lower concentration. So next, reinjection reproducibility. So when we are repeatedly doing the experiment, same injection, same sample, you should get the same result, not different result. So that results, acceptance criteria are given. Okay. So next, stability. So stability will be chemical stability as that of the similar one. And in this thing, to establish a stability, they'll be using the freezing and thawing, means 3 cycles. First freezing, and then again normal temperature, then freezing, normal temperature, then freezing, normal temperature, so that it is a cycles will be followed. And then benchtop stability, how much time it is stable in the table, and then stock solution stability, because this data is very important. Once if you are going for any new analytical method, when you are doing the experiment, you can fix the time. For example, when you do the experiment, you may take half an hour, you may take 1 hour, you may take 1.5 hours also sometime. So how much time the solution is stable? Once if you record it, then they will follow the same thing, so that we can have a idea within up to 1 hour this sample is stable, so that we can go for the scanning within 1 hour. Sometimes some drugs will be very sensitive, it will be stable for only 10 minutes or 15 minutes. So in that condition, we have to fast our work, we have to fast our scanning time, and we have to finish within half an hour. So that is the reason, because nowadays the people are doing research without knowing the reason. Why we are doing this stability data, why we are doing this one, they keep on doing whatever their elder did. So that is the thing. So you have to follow the regulation. Stock, long-term stability, process sample stability, all those things. Next. Thank you. So with this I conclude my lecture. And I hope it may be giving you little spark in your area. If any discussion, welcome. First of all, I thank organizers. I thank all the 3 juries and other people, those who listen here. Thank you, one and all. Any discussion you can have.
Thank you for your insightful discussion. Your expertise and perspective added depth to our understanding of the subject. Let's move on to the interactive session.
Mic என்ன கொடுங்க.
You are giving a step by steps. PK methods are there.
Which?
PK methods.
Pharmacokinetic method.
PK method.
Pharmacokinetic method.
Pharmacokinetics. Okay. Thank you, sir.
Any online people? Sure, it is there with thing. Actually, everything is available in USFDA website and ICH guideline. I took from the ICH guideline. I took from the USFDA guideline. So you can get it from, even PowerPoint is available. It is not secret. It is open to all. I have given a PowerPoint to the organizers. You can collect from the organizers. No problem. Any question from the student side, I expect?
Any questions from the student side?
I invite Dr. Surendra Kumar sir to share a few words about the session.
Good afternoon. The lunch time la ivla pormiya ukandinga kettadhukku romba nandri. Sir, it's a really wonderful session. In the lunch time la ivla busy engage பண்ணி வச்சிக்கிறதே பெரிய விஷயம். அது ரிமார்க்கபிளா இருந்துச்சு. ரெகுலேட்டரி ஆஸ்பெக்டா இருக்கட்டும், இல்ல ஃபேஸ் ட்ரையல்ஸ் பத்தி நீங்க கொடுத்ததா இருக்கட்டும், இல்ல பயோமெடிக்கல் வேலிடேஷன்ஸ் பத்தி பேசுறதா இருக்கட்டும், எல்லாமே ரொம்ப நல்லா இருந்துச்சு சார். On behalf of the Shanmugha College of Pharmacy and participants, I personally thank you, sir. Thank you once again, sir.
I would like to invite the other two chairpersons to have few words about the session.
So good wishes to one and all present here. First of all, I thanks to this management and program coordinator and the teaching staffs and all the faculties and all the committees inviting me for as a chairperson for this wonderful occasion. So I know Kannappan sir. So personally I know he have a good character, behave, act and everything. So whatever we need in the pharmacy field, whenever we ask, without hesitation, sir always be help for me. Sir is also my guru for both M.Pharm as well as PhD. He is guiding more for me. So I thanks to Dr. Kannappan sir. So we are very grateful thanks to today's chief guest Dr. Kannappan sir who have delivered his valuable and delightful speech on the topic. I think everyone we get the feed of knowledge from our sir's speak on this topic. So sir is clearly explained about clinical trials, pre-marketing surveillance, post-marketing surveillance, drug development process, functions of regulatory affairs development, drug master file and different types of drug master file, new drug applications and upgradation of new drug applications and CDSCO functions, and also sir is clearly explained about bioanalytical method validation and parameters. Once again, we thank our chief guest Dr. Kannappan sir for delivering his valuable speech and delightful and knowledgeable speech for us. Thank you, sir.
Good afternoon to all. Dr. Kannappan sir was clearly explained about preclinical trials and clinical trials and what are the phases are there in the clinical trials and how to get the approval, how long it would take for approval of new drug. I think almost 14 to 20 years. And sir was explained about drug master file. It's very most important one. And also for sir was given one new bill. It was introduced in 2022, new drugs and medical devices about that. Sir, I have one doubt about that, whether it was introduced or it's going to introduced. Still it not introduced. It's most important one. Students, you will get more job opportunities if the bill was introduced. You will get very job opportunities in MNC itself. Okay, that very informative to all, sir. And sir was explained about regulatory affairs. The UG students have the syllabus in the 8 semesters only. That also for elective subjects. It's most informative about the regulatory affairs. It's very nice, and very thank you so much, sir.
I am pleased to invite Dr. K.K. Sendil Kumar Sir, Professor and Head of Pharmaceutical Analysis Department to honor our prestigious chairperson, Dr. Surendra Kumar Sir. I kindly invite Dr. C. Manikandan, Professor and Head of Pharmacology Department to honor our chairperson, Dr. Mohanraj Subramaniam.
Now I invite Dr. S. Anand Kumar, Professor and Head of Pharmacy Department, to honor our prestigious chairperson, Dr. Rajarajan. Now I kindly invite all our chairpersons to honor our chief guest, Dr. N. Kannappan.
Yes. Thank you all for your patience. Lunchtime is not just a pass in a day. It's an opportunity to fuel up for success. Enjoy your meal and come back ready to conquer the afternoon's session. Lunch is served at H Block. I request all should gather here back at 2:55 p.m. Thank you. Welcome once again. Let us make this conference an unforgettable experience for all.
It's my distinct pleasure to extend a warm welcome to Dr. M. Senthil Kumaran, M.Pharm, PhD, our esteemed chairperson for this conference. We are truly honored to have him join us in guiding the proceedings of the event. Dr. M. Senthil Kumaran brings with him a wealth of knowledge, expertise, and leadership that will undoubtedly enrich our conference experience. His dedication to his field and his passion for fostering collaboration and innovation make him an invaluable addition to our team. So without any further ado, let us extend a warm welcome to Dr. M. Senthil Kumaran and express our sincere gratitude for his willingness to lead us to the journey of discovery and collaboration. great pleasure, I welcome you to the 3 days National Conference on Recent Innovations in Pharmacological Research 2024. Today, we are privileged to have Dr. J. Saravanan, M.Pharm, PhD, Assistant Professor of JSS College of Pharmacy, Tamil Nadu. We are deeply grateful to Dr. Saravanan for taking the time out of his very busy schedule to be with us today. His presence here serves as a testament to his commitment to the event. Let me kindly invite Dr. M. Senthil Kumaran to have a few words about our chief guest.
Good afternoon. Good afternoon, one and all. So I am very happy to participate in this wonderful event. First, I would like to thank the management, principal, and the organizing committee of this wonderful. The 2 days back, I was very happy when I was got a call from this your staff, Mr. Gopal. He invited me for that chairperson. Well, because very happy regarding today's special guest. Hello. I'm very happy to participate in this wonderful event. The 2 days back, very happy because today's special guest, Dr. Arham sir, he was my vice principal when I was in KMCH. So anyhow, we are going to meet this Dr. Arham but unfortunately he left this morning afternoon. And other happy to introduce this today's speaker, Dr. Saravanan. I know him in the last 15 years as a faculty in the KMCH College of Pharmacy. Welcome you, sir. And other happiness and before introduce that speaker, this morning today's chief guest Dr. Arham and today's speaker Dr. Saravanan have one similar character because I know him for 15 years and I know him the chief guest Dr. Arham last 15 years. Both are very cool person. I never seen those are agitated or in the emotional mode in the last few years. So the same we are expecting this afternoon, sir, from your speech. You said we after had a very good lunch. Yes, we had a very good lunch. Also, we are going to get a very good thought from our speaker, Dr. Saravanan. Saravanan sir, we are expecting only. Afternoon we had a very good lunch and happy. The same we are expecting from your thought. The next 45 minutes. And Dr. Saravanan, he was completed his B.Pharm in the year of 2006 from Chennai College of Pharmacy, Keynote. Followed by he was did his M.Pharm, Department of Pharmacology in KMCH College of Pharmacy in the year of 2009. Followed by he was completed his PhD in JSS College of Pharmacy in the last year 2022. Now presently working as a professor in JSS College of Pharmacy and examined in the various universities. So many things I have to thought about discuss about that or tell about Dr. Saravanan, but due to lack of the time, I will complete and I will welcome Dr. Saravanan to present your valuable thoughts. Thank you. Thank you so much.
Thank you, sir. Let's have a short AV, please.
It's my pleasure to introduce Dr. J. Saravanan, a distinguished educator and It's my pleasure to introduce Dr. J. Saravanan, a distinguished educator and researcher in the field of pharmacology. Dr. Sharavanan's journey in academia has been marked by unwavering dedication and profound contributions to the advancement of pharmaceutical sciences. With a solid educational foundation, including a bachelor's degree in pharmacy from Chennai College of Pharmacy and a master's degree from KMCH College of Pharmacy, Dr. Sharavanan's pursuit of knowledge led him to earn a PhD from JSS College of Pharmacy, UT. His doctoral research focused on exploring new frontiers in pharmacology, paving the way for groundbreaking discoveries in the field. Throughout his illustrious careers, Dr. Saravanan has served as an assistant professor at esteemed institutions such as JSS College of Pharmacy, KMCH College of Pharmacy, and AKRG College of Pharmacy. His commitment to excellence in teaching and mentorship has left an indelible mark on countless students, shaping the future of pharmacy education. In addition to his teaching responsibilities, Dr. Saravanan has made significant contributions to pharmaceutical research as evidenced by his numerous publications in prestigious journals. His work spans a wide range of topics from the inhibition of calcium-forming oxalate to the therapeutic potential of novel drug candidates in various diseases. Dr. J. Saravanan has made significant contributions to the field of pharmacology through his major publications and professional roles. His research has led to the publication of several impactful articles in renowned journals addressing critical issues in healthcare and pharmacotherapy. Beyond his research endeavors, Dr. Saravanan has also played an important role in academia as an external examiner and question paper setter for pharmacy programs at various universities. His commitment to advancing pharmaceutical sciences through research, teaching, and academic leadership is truly commendable and serves as an inspiration to his peers and students alike. His dedication to ensuring the quality and integrity of pharmacy education is evident through his involvement with prestigious institutions such as the Tamil Nadu Dr. M.G.R. Medical University in Chennai, Kuhas in Kerala, Vinayaka Mission University in Salem, and few others. As an external examiner, Dr. Saravanan has played a crucial role in evaluating and maintaining the standards of B Pharmacy, M Pharmacy, and Pharmacy programs at these estimated universities. His insightful assessments have contributed to the enhancement of academic excellence and the development of competent pharmacy professionals. Furthermore, Dr. Sharavanan's involvement as a question paper setter reflects his commitment to crafting assessments that accurately gauge students' knowledge and understanding of pharmaceutical concepts. Dr. Sharavanan's multifaceted distinct contributions to pharmacy education underscore his dedication to nurturing the next generation of pharmacists and ensuring the continued advancement of the pharmaceutical field. His exemplary service and expertise has undoubtedly left a lasting impact on the academic community and will continue to inspire excellence in pharmacy education for the years to come.
please join me in extending a warm welcome to Dr. J. Saravanan as we eagerly anticipate their insights and perspective on network pharmacology.
Thank you, sir, for your kind words. Good afternoon, all. It's a pleasure to be here this afternoon. Okay. Okay. Good afternoon, all. It's a pleasure to be here this afternoon at Shanmugha College of Pharmacy. I thank the organizers for having me here to deliver a talk on network pharmacology. Thank you all. I split my talk on network pharmacology into two parts. In part one of this lecture, I'll explain the basics of network pharmacology. And in part two, I shall deal with the general methodology of network pharmacology. Let's get started with part one. Arham's law is an observation that discovery of new drugs is becoming slower and more expensive, despite the advancements in technology like high throughput screening, computer-aided drug discovery, and combinatorial chemistry. Using these techniques, we can synthesize and screen millions of compounds in a very short period of time. In general, when technology improves, the output also improves. But this is not the case with drug discovery. The technology has improved a lot, but discovery of new drugs has remained more or less the stagnant. There are four main reasons for this disappointing trend in discovery of new drugs. The first reason is the cautious regulator problem. No government wants a thalidomide-like disaster again under their watch. So discovery of new drugs is becoming tougher and tougher. It is very difficult these days to get past the regulator. The second reason is an increase in cost of drug discovery. The prices are increasing. We have to spend billions of dollars to come up with a new drug. The third problem is a difficulty in discovering new drugs that are better than the existing drugs. For example, it would be very difficult for us to come up with a new statin that is better than Lipitor. If a pharmaceutical company spends billions of dollars in discovering a new statin, the company would definitely expect the new statin to be better than Lipitor. If it is not going to be better, it is going to be a huge setback for pharmaceutical companies. iPhone 16 should definitely be better than iPhone 15. If not, there is no incentive for discovery or innovation. But these three reasons are not the most important reasons for a slowing down in drug discovery. The most important reason is a reason called the basic research brute force bias. The basic research brute force bias is based on the hypothesis that high affinity binding of a single ligand to a specific target linked to a disease would offer significant therapeutic benefits. Of course, this approach works fine with simple monogenic disorders like sickle cell anemia, hemochromatosis, Tay-Sachs disease, and hemophilia, etc. But when it comes to complex diseases This target-based approach, this single ligand, single target-based approach does not offer much results. Okay. But complex disease results from a combination of multiple genes, environmental factors, and lifestyle factors. Complex diseases involve multiple genes as well as their interactions with lifestyle factors. Examples of complex diseases include atherosclerosis, hypertension, diabetes, Alzheimer's disease, and cancer. Human physiology is complex. Human pathology is even more complex. We cannot restrict a complex disease to a single target. The idea of magic bullets is great, but in practice, it's probably not going to be the right approach for complex diseases. So we need to explore other approaches in the field of drug discovery. One such approach is network pharmacology. Network pharmacology uses two or more drugs acting on multiple targets involved in a disease-causing network of proteins. This sounds like combination therapy, but network pharmacology is not combination therapy. In combination therapy, we use two or more drugs that act symptomatically on unrelated targets. Whereas in network pharmacology, we use low doses of two or more drugs that act on multiple closely related proteins involved in the pathogenesis of a disease. I hope that is clear. The concept of network pharmacology, that is the concept of drugs acting on multiple receptors or multiple targets, is not entirely new. Even some existing drugs act on multiple receptors. These drugs that act on multiple receptors, I mean the existing drugs that act on multiple receptors to produce a pharmacological action, are called dirty drugs. Sunitinib, an anti-cancer drug, is a multiple tyrosine kinase inhibitor. It does not act on a single receptor. It acts on multiple receptors like vascular endothelial growth factor receptors, platelet-derived growth factor receptor, CD117, RET receptor, CD114, and CD135. Clozapine, a dopamine receptor antagonist, not only acts on dopamine receptor, but it also acts on serotonin receptors, adrenergic receptors, muscarinic receptors, etc. But clozapine's action on these additional receptors leads to unwanted adverse effects. But clozapine also acts on PPAR receptors. Clozapine's action on PPAR receptor is believed to be a contributing factor to its therapeutic benefit. There is one more anti-cancer drug called sorafenib. It is also a multiple tyrosine kinase inhibitor. It acts on multiple receptors like RAF1, BRAF, c-KIT, FLT3, VEGFR2, VEGFR3, and PDGFR-beta. Other examples of dirty drugs include tramadol, chlorpromazine, olanzapine, dextromethorphan, ibogaine, kanamycin, and axitinib. Before we proceed further, we have to understand how to interpret a network. It's very simple. It has only two things in a network. There are only two things in a network: circles and lines. The circles in a network are called nodes. The lines in a network are called edges. The nodes represent proteins. The edges represent the association between proteins. The colored nodes, the nodes in different color in a network, represent the first level of interactors. White nodes represent second line of interactors. Edges come in different colors. For example, the dark purple colored edges in a network represent those associations that were determined through experiments. The yellow colored edges represent associations that were determined using text mining. So they come in different colors. It's very straightforward. You can go through the legend. The filled nodes represent a protein with known 3D structure. An empty node represents a protein with unknown 3D structure. I hope this is clear. If this is clear, we'll move on to the second part of the lecture, the general methodology of network pharmacology. This is the figure that represents the general workflow of a network pharmacology study. There are only 8 steps and it is very simple. The first step includes choosing your ligands and a disease of your interest. So the first step is to choose your ligands. The second step is to predict the targets that our ligands can act upon. We can use databases like Swiss target prediction, Stitch database, and binding DB database to predict the targets that our ligands can bind to. The third step is to identify the targets involved in the pathogenesis of our chosen disease. For example, if you have chosen Alzheimer's disease, you have to identify the targets that are involved in the pathogenesis of Alzheimer's disease. There are several databases that will help us to do it. A few important databases are DisGeneT, GeneCart, and Therapeutic Target Database. The fourth step is to identify the common targets. Common targets are those targets that can bind to our ligands and are also involved in the pathogenesis of our chosen disease. For example, in this picture, 994 proteins bind to our ligands and 719 proteins are involved in the pathogenesis of, that is, the Alzheimer's. Now we have 135 proteins that can bind to our ligand, and those same 135 proteins are also involved in the pathogenesis of Alzheimer's disease. So we have 135 common targets. The next step is to find out whether these proteins have any associations between them or not. If there are associations, that is fine. Not all proteins will be associated. Most of the proteins will have some kind of association between them. Now we have 135 proteins. We cannot work on 135 proteins. Not all 135 proteins in a network would be equally important in a signaling network. A few proteins might be highly important, whereas others might play only a minor role. So our next step is to identify the key proteins in a network. To identify the key proteins in a network, we have to do topological analysis. So topological analysis will analyze the network and it will inform you about the key proteins involved in the signaling network. Now we have a set of proteins, but we basically do not know what these proteins do. We have to know the function of these proteins, what they do in a cell, where they are located, what microfunction they do, etc., what molecular function they do. In order to find out the functional role or the physiological role of a protein, we have to perform gene ontology studies. And we should also know which proteins are involved in which pathway in a disease. A disease is not caused by a single pathway, there might be multiple pathways. So we have to find out in which pathway, in which biological or disease pathway our proteins are involved. To find that out, we have to perform a pathway analysis. Next, we have to perform in silico studies. We can do ADMET study, we can do drug likeness study, and we can do molecular docking and molecular dynamics study to find out the best ligands. The last but the most important step in network pharmacology is biological validation. We have to validate our results using in vitro and in vivo studies. I hope this general outline is clear. I'll explain each and every pathway in detail. So the first step is to choose ligands. You can synthesize a series of compounds and work on it. If not, you can choose an existing drug or a group of 1 to 3 existing drugs and work on drug repositioning using network pharmacology. If not, you can work on natural products. In the first two cases, we do not have any problems in choosing the ligands because we are going to synthesize the compounds and we are going to work on existing drugs. But if you are working on plants, we should know what phytoconstituents are present in those plants. To do that, we have two databases. These are online databases and it's free. We can use it. The first database is called Impact Database. Impact stands for Indian Medicinal Plants, Phytochemistry and Therapeutics. It's an online database that provides information about Indian medicinal plants. Use any browser and access this web page. The home page would look somewhat like this, so that is the first step. In the home page, you will find a tab, Basic Search. Hit on the Basic Search button. It will take you to the next page with a search box in it. In the search box, type the plant or plant name of your interest. It could be any plant. If you type the name of your plant and hit the search button, it will give the list of phytochemicals present in different parts of a plant. Download the results and save it. There's one more database called Dr. Duke's database. This also provides information about medicinal plants. So using your browser, access the home page of Dr. Duke's database. The home page would look like this. And in the home page, you will find a search box. Before you type the name of your plant, there is something called all entities. Hit that box and choose plant. After you choose plant, type the name of your plant. After typing the name of your plant, hit the search button. It will also give you the list of phytoconstituents present in different parts of a plant. Sometimes you may not find your plant in Impact or Dr. Duke's database. In that case, you have to do a manual search. You have to search scientific databases like ScienceDirect, PubMed, and Google Scholar and find relevant literature. If literature is available and if you are able to collect a considerable number of ligands, it's fine. If you are not able to find ligands, if you are not able to find any literature on your plant, then you are left with only one choice. The only choice is to do experiments on your plant and find out the phytoconstituents. Okay, we cannot proceed further in network pharmacology with common names. We cannot use words like routine, mangiferin. We cannot use words like aspirin, diclofenac. You cannot even use iupac name. We have to convert these common names into a format called SMILES format. To do that, you have to visit a web page called PubChem. In the home page itself, you will find a search box. Type the name of your ligand. It could be routine, mangiferin, levodopa, or whatever it might be. Hit on search. You'll get a very long list. You'll get many results. All those results are not relevant to us. The first thing that is relevant to us is 2.1.4. In 2.1.4, the structure of a ligand will be converted into a format called SMILES format. SMILES format is like a formula that represents a chemical structure. The next important thing that we need is, so the first we need SMILES format. The second thing that is important is 3D structures of your ligands. To get that, click on structures. You will get a 3D structure, and on the top you see a download button. Hit on the download button, it will take you to the next page. The structures are available in different formats, but what we are interested is in SDF format. Okay, so click on save and save the structure in SDF format. These structures would become helpful when you move on to in silico studies. I hope that's clear. You can save the structure either in 2D format or 3D format. The third step in network pharmacology is to identify the disease targets. There are several databases that do it. The important databases are Swiss target prediction, Stitch database, and binding database. These databases predict the probable proteins or targets based on the principle of chemical similarity. The logic here is that if two drugs are similar in structure, they might bind to similar proteins. So using these databases, we can find out targets. Okay, so the first page, database is Swiss target prediction. Use any browser, type Swiss target prediction. Most often, it would be the first hit. Click on it, it will take you to the home page of Swiss target prediction. Okay, now next we have to select the species. We can select Homo sapiens or rat or mouse, whatever it is. Under that, you'll find a box. In this box, you have to paste the SMILES format of your ligand. After pasting the SMILES format of your ligand, click on the predict targets button. Once you do it, it will give you a list of proteins that your ligand might probably bind to. Okay? It's just a prediction, so it might give you a list of proteins like this. So the first receptor is G protein coupled receptor. Apart from the name, it would also give you the UniProt ID, to which class of receptors it belongs to, and the strength of probability. In the first box, the green color is complete because the prediction is almost 100% correct. But when you go down the list, the strength of prediction decreases. You can select only the first targets. If not, you can select all the targets. It depends on your choice. You can set the probability limit even before you start your study. Stitch is also a similar database that can be used to predict the targets that your ligands act upon. So type Stitch in any browser. The first hit most probably would be Stitch. Click on it, it will take you to the home page of Stitch database. In the Stitch database, click on item by name. You can also enter multiple names, but it is better to do one item at a time. In the search box, enter your ligand of choice. It can be magicirin or aspirin or diclofenac or whatever it is. Choose the organism as Homo sapiens. If you are interested in rat or mouse, you can choose those species. After that, you can hit search. Once you hit search, you will get a list of targets that your ligand can act upon. For example, magicirin can act on these receptors. On the right end, you'll see some numbers, 0.938. It means that the first prediction is 93.8% accurate, but the last protein is only 80% accurate. Again, you can set the probability limit and you can select the proteins. The last database is called Binding DB database. Type Binding DB in any search button. After that, click on the first hit, it will take you to the home page of Binding DB. On the left side, you'll see a tab called Special Tools. Click on it, And under special tools, you see an option, find my compounds target. Click on it and again enter the SMILES format. In Stitch, you can enter the common name, but in binding DB, you have to enter the SMILES format. Enter the SMILES format. You can also set the probability limit over there. In similarity, it's 0.85 by default. If you want, you can change it to 0.9 or 1, etc. Click on Go. You will get a list of proteins that our ligands can bind to. Now we have a set of proteins that our ligands are capable of binding to. Our next step is to identify the targets that are involved in the pathogenesis of a disease. There are 3 important databases to do it. The 3 important databases are DisGeneT, GeneCards, and Therapeutic Target Database. Type DisGeneT in any browser. The first hit would most probably be DisGeneT. Click on it, it will take you to the home page. In the home page, click on the search button and type the name of your disease and hit the search button. You will get results like that. And under the results, there is a summary of gene disease associations. If you click that button, it will give you an exhaustive result of proteins that are involved in the pathogenesis of Alzheimer's disease. For example, DisGeneT provides a list of 3,397 proteins that are involved in the pathogenesis of Alzheimer's. The strength of probability keeps on decreasing as we go down. If you set a high probability value, the number of targets would decrease. The choice is ours. The next database is called GeneCards database. Type GeneCards in any search engine. The first hit will be GeneCards. Click on GeneCards. It will take you to the home page of GeneCards. In the home page, you will find an option called MalaCards. Click on MalaCards. It will take you to the next page. In the next page, you will see a search box. In the search box, enter the name of your disease that you are interested in and hit the go button. Once you do that, you will get a list of proteins that are involved in the pathogenesis of that disease. Okay, the next database is Therapeutic Targets database. I made a mistake here. It's GeneCards in the slide name. It should have been Therapeutic Target database. So go to the home page and in the search box, type the name of your disease that you are interested in, and it would provide you with the proteins that are involved in the pathogenesis of that disease. So now we have disease targets and we also have ligand targets. Now all these targets are not equally important, so we have to find out the common targets. We have to find out those targets that can bind to our ligands and they can also involve in the pathogenesis of that disease. To do that, there is a tool, there is an online tool called Winnie. Go to that website and in the list 1, enter the name of all ligand targets. And in the list 2, enter all the disease targets and click enter. Once you click enter, you will get the result. The results will include only the common targets. Common targets are those targets that can bind with our ligand and are also involved in the pathogenesis of that particular disease. So at the end of this study, we might have 44 targets that are involved in the pathogenesis of a disease, and those targets also bind to the ligand. Now we have around 44 proteins. All 44 proteins might be associated with each other or not. So we have to confirm whether these proteins have any relationship between them or not. To do that, there is a database called string database. So use any browser and type string and click on the first hit. It will take you to the home page of string database. In the string database, in the home page of string database, click search. It will take you to the next page. In the next page, click multiple proteins. In the box, enter the list of all 44 proteins, all 44 common targets. Select organism as Homo sapiens and hit search button. It would give you a network like this. Okay? As we discussed earlier, the circles are called nodes and they represent protein, lines are called edges, and edges represent associations between the proteins. So as you can look at the end of that slide, there are some proteins like AMY1C and FTO. These proteins are not associated with any other protein. So those proteins are not very important. Now we still have somewhere around 40 proteins. We cannot work on 40 proteins. It would be very difficult for us to come up with a ligand that acts on 40 different proteins. So we have to narrow down the list furthermore. To do that, we have to perform something called topological analysis. The aim of topological analysis is to identify the key proteins in a network. So we have many proteins in this network. If you do a topological analysis, we can find out the top 10 key proteins or top 15 key proteins, etc. So we use three measures to do topological analysis: degree centrality, closeness centrality, and betweenness centrality. Okay. In Tamil Nadu, we have airports in Chennai. We have international airports in Chennai, Madurai, Tiruchi, etc. But which one do you think is the most important international airport? Which airport do you think has the most connectivity? Obviously, it is going to be the Chennai because it has high connectivity. You can travel to any part of the world from Chennai International Airport. So a protein that has maximum number of association with other proteins has the highest degree. Okay, fine. So Chennai is the most important airport. Similarly, a protein should be important in a network because it is connected to many proteins in that network. So when you measure degree centrality, you will know how many connections a protein has. The results would be somewhere like 6, 9, 10, etc. A protein with a degree value of 10 is better than a protein with a degree value of 6. Okay, well, the next parameter that we measure is called closeness centrality. Chennai is not the closest airport to all countries in our world. The closest airport to all the countries in our world should be in the middle of the network. For example, the red colored nodes in that figure is closest to all other nodes in the pathway. The next one is called betweenness centrality. Flights from Chennai do not leave to Europe directly. They might land somewhere for transit. Most often it is going to be Dubai. So what do you think will happen if there is a problem in Dubai airport? Will Chennai airport be affected or not? If flights from Chennai cannot leave to Dubai because there is a problem in Dubai, what happens? There will be problems in Chennai airport as well. Okay, fine. So betweenness centrality measures the most important or critical node in a network. So by measuring these three parameters, we can find out the top proteins in a network. To do that, we have to download a software called Cytoscape. It's a free software, you can download it. When you develop a protein-protein interaction using string database, at the bottom of the network, you'll find an exports tab. Click on that exports tab. When you click on that exports tab, you'll see an additional option at bottom, send network to Cytoscape. Click on that button. The protein-protein interaction network will be exported to the Cytoscape database. Once you have the protein-protein interaction network in the Cytoscape, click on Tools. When you click on Tools, you'll get an option, Analyze Network. Click on Analyze Network. On the right side of the page, you will get a summary of your protein-protein interaction network. It will give you the total number of nodes, total number of edges, etc. But we are not much interested in those things. What we are interested is degree centrality, closeness centrality, and betweenness centrality. At the bottom of this page, on the same page, you will find the results of betweenness centrality, degree centrality, and closeness centrality. So we started with 44 proteins. We cannot proceed further with all 44 proteins. You can narrow down the number of proteins to 10. You can select 10 proteins with better degree centrality, closeness centrality, and betweenness centrality. So once you have a list of 10 proteins, you can move on to the next part of network pharmacology. The next part is gene ontology. We need to have an idea about the functions of those selected proteins. If the function of those proteins that we selected is not in any way related to the disease that we are studying in, that protein is no longer useful. You can discard that protein. When you do a gene ontology study, it will give you the molecular functions of that protein, biological process of that protein, and cellular component of that protein. For example, if you are dealing with hemoglobin protein, it will tell that the biological process of hemoglobin is to transport oxygen. The molecular function of hemoglobin is to bind oxygen. And it will also tell where in the red blood cell hemoglobin is located. It will tell us whether the protein is located in cytoplasm or in the nucleus, or what other structures it is associated with. This study will help us to further narrow down the list of proteins. To do gene ontology, we use an online tool called ShinyGo. Go to the homepage of this website and type the name of your proteins and click submit. When you click submit, you will get results of biological processes of that protein, molecular function of those proteins, and cellular component of the proteins. For example, the proteins that I have entered are mostly associated with chromosome and chromatin in the cell. So it would give us a general idea about the importance and location of a protein in a cell. Next, we also have to identify in which pathways our proteins are involved in. To do that, we have to perform a pathway analysis. We use a tool called Reactome to do pathway analysis. Go to the homepage of Reactome website and click on Analysis Tools. It will take you to the next page, and there is an empty box. You can enter the name of all your proteins in that box and click Continue and click Analyze. When you click Analyze, it will give you a list of pathways that our proteins are involved in. If you find out that particular pathway is also involved in the pathogenesis of our disease of interest, it means that we have done a good job. The number 4 in the first box, in the second column, says that out of the 10 proteins that I entered in the search box, 4 proteins are involved in that pathway. It would also give you a pathway result. You can also download the result by clicking on downloads. You will get somewhere around 50 to 60 page PDF. You can read it. You will get a lot of information from that result. So we started with somewhere around 44 proteins. Now we have only 4 important proteins. So now we can work on these proteins further. We have narrowed down the number of proteins, but we have not narrowed down the number of ligands. For example, if you are working on a plant, there might be hundreds to thousands of phytoconstituents present in a plant. We cannot work on all those ligands. We have to decrease the number of ligands. To do that, we can perform an ADMET analysis and drug likeness study. This study can be followed by molecular docking and molecular dynamic study, in vitro studies, and in vivo studies. If you want to do an ADMET study, use an online tool called ADMET. Go to the home page of Admit 2.0 and click on Admit Screening. If you are working on a single ligand, click Admit Evaluation. If you are working on multiple ligands, click on Admit Screening. Click on Admit Screening, it will take you to the next page. Copy and paste all the smiles. You can paste up to 500 ligands, the smiles of 500 ligands at one time. Click Submit. It will give you the results of Admit studies of each compound. If you want to view the results of the first compound, click View. If you click View, you will get the physiochemical property of your ligands, all physiochemical property of your ligands. You will get drug likeness study like Lipinski rule, Pfizer rule, GSK rule. You will also get predicted toxicity results. You will get information related to absorption, distribution, metabolism, excretion, etc. If you are working on a central nervous system disorder, and if your ligand is not able to cross the blood-brain barrier, there is no point in working with that ligand. For example, in this result, the drug, the ligand does not cross the blood-brain barrier. If I am working on Alzheimer's disease, I'll skip that ligand. So by analyzing the physicochemical properties, medicinal chemistry properties, pharmacokinetic properties, I can narrow down the number of ligands. These studies can be further followed by in silico studies like molecular docking and molecular dynamics. I don't have time and it is out of the scope of my discussion, so I will not be discussing in silico studies. In silico studies must be followed by biological validation. To validate your results, you have to perform in vitro and in vivo studies. Network pharmacology is not perfect. It has limitations. The first limitation is data integration and quality. Most of the data that we obtain are predicted from experiments that were done by someone else. We do not know the quality of those experiments. We did not do the experiments. We got the results from someone else's experiments. We do not know how good those results are. The second limitation is biological complexity. Human body is very complex, physiology is complex, pathology is complex, so we cannot just predict things. We have to do experiments. The next one is predictive accuracy. We do not know how accurate the predicted results are. And the last disadvantage is a lack of standardization. I will go through the procedure in a short way, just to recap. So the first step is to select ligands. The ligands can be synthetic, semi-synthetic or natural. Identify the ligand targets. Identify disease targets. Next, select the common targets. Perform network analysis and topological analysis. Perform gene ontology and pathway analysis, followed by pharmacokinetic studies, in silico studies like molecular docking and molecular dynamics, and finally do biological validation. Once you do biological validation, it will add value to your network pharmacology resource. The most important problem with the current approach in drug discovery is we work on a single ligand and a single target. 20 years down the line, this approach has not given us significant results. So we have to explore other areas. In most cases, especially in complex diseases, a disease is not caused by a single protein, but by a network of proteins. So it is better to work on ligands that act on multiple proteins rather than a single protein. And the last thing is effective targeting of multiple targets using network pharmacology approach might even help us to cure diseases that are incurable currently. These are some of the publications. And thank you.
Thank you, sir. Your session has been both enlightening and inspiring. Is there any questions from student side? Now I invite both the chairpersons to share a few words about the session.
Good evening, sir. I am Shivagita from Erode College of Pharmacy. I have a doubt. There are a lot of softwares for identifying the protein targets. So there are many targets for identifying the protein targets. So which one to rely on?
There are even more databases, so it is up to us. It's a personal choice. What you feel comfortable with. If you're comfortable with binding DB database, you can use it. If you're comfortable with Stitch, you can use it.
Is there any standardization?
No, there is no standard method. It's a personal choice. It's up to us.
Okay, sir.
You can work on all three databases. You can get the results. You can compare the results, and one that satisfies you the most, you can proceed with that one.
Okay, sir. And one more doubt regarding ADMET analysis. So other than Admit 2.0, any other softwares to do Admit analysis, sir?
There is also a Swiss database for pharmacokinetic parameters, and there are many others. Right now I couldn't recall, but the most widely used is Admit. You can get a lot of results.
Okay.
You can get medicinal chemistry results, you can get drug likeliness, and you can get pharmacokinetic parameters, etc. And the most standard one would be Quick Pro from Schrodinger.
Okay, sir. Thank you. Thank you.
Anyone else? Ali Malarkudi ma'am, please share a few words.
Good evening to one and all present over here. First of all, I would like to thank the principal and the management of Shanmugha Educational Institution for giving me this opportunity. This session, I think it was a very wonderful session. Theoretically, everybody, I think students they know what is meant by network pharmacology, but practically, I think they lack in that particular topic. And Dr. Ceremonial has given me a crystal clear practical issues in this topic. It is nothing but a computational approach. It highly ascertains the therapeutic mechanism of drug for targeting the disease at the level of biological targets and pathways. Because no drug therapy without a protein binding. Maximum of drug is mainly based on the protein binding, and the therapeutic efficacy can be usually based on the protein binding. So the selection of a protein and the selection of a target and selection of the drug is very, very crucial. So based on this talk, we can try to reduce the adverse effect and side effects, and we can increase the therapeutic activity of the drug. So it mainly concentrated on the compound target and the diseased genes. So you heard about so many databases which is available, so many softwares which is available in the net. We can, he has given a list of softwares. But I would like to ask one thing from Surrounding. Sir, can you suggest any of the practical or the practice can be given by any of online way, any thing is available for this for students, sir? Okay. Okay, so because the students doesn't know how to approach this kind of a software and how to work on it, This is very, very crucial for the students. So please give some kind of guidance to our students. Anyway, your talk is very, very useful for the students. Thank you so much, sir.
I kindly invite our board chairpersons to honor our beloved chief guest, Dr. J. Saravanan.
I would like to call upon stage Dr. P. Perumal, Vice Principal, Sri Shanmugha College of Pharmacy, to honor our chairperson, Dr. M. Senthil Kumar. I would also like to call upon stage Mrs. N. Vinodini, Professor, Department of Pharmaceutics, to honor our chairperson, Dr. Ali Malarkodi. Thank you all. Now the session is open for oral presentation.
Presentation.
I would like to invite Dr. S. Alimalar Kodi and Dr. J. Saravanan for oral presentation evaluation. Now we have our first oral presenter, Mr. John Ankumani from Annamalai University.
Good afternoon, everyone. My research topic directly go to the topic anti-diabetic, anti-hyperlipidemic effective of lactic acid and streptozotocin induced diabetic rats. My name is John, research scholar in biochemistry, Annamalai Universities. My guide name is R. Kausalya ma'am. Next. The objective is first we introduce about the diabetics, type of diabetics. Next, diabetic testings, symptom of diabetics, prevention and strategies, effectiveness of diabetics. Next, management diabetics. Next, the global diabetics. Yeah. In 2004, when compared to study for 2021, the diabetic is a chronic metabolic disease characterized by evaluations level of blood glucose, blood sugars, which leads to times serious damages to the heart, blood vessels, eyes, kidneys, and nerves. For the people living in diabetics, most stressful supportable treatment, including insulin, in critical steps available globally. Agreed to target the health diabetics and obesity by 2025. About 422 million people worldwide have diabetics. The major living attributes to diabetes each year. Both the number of cases and prevalence and diabetics strategies increase in these patients. Next. In Tamil Nadu, Makkalai Thedi Marandam scheme, this launched in 2021. They have first identified this year, 2022 to 24, directly to visit the primary health centers to identify the patients, hypertensions, and along with the diabetic patients. But in this year, we know about this 2,750,000 people for more diabetic identified, and couple of months to find out this periodical checkup. For this year, such much of our rate was 3,12,000 people, moreover the diabetics, and along with the hypertensions, and then normal to the others. Next. In this diabetics, I have two types. We have find out these. Yeah. Two types, type 1 and type 2. What is diabetics? Do you know about this? Chronic. Okay, next question is that's it. Conditions which the body glucose do not consume of the glucose. Type 1 and type 2. The level of insulin is decreased. This is a type 1. The type 2 is enough insulins and other resistant to the axons. The more predominant the type 1 and type 2, these autoimmune conditions leads to insufficiency of the form pancreas for the type 1, and they always required for the insulins given by multiple daily injections via insulin pump. Do you know about this? Next ones. Which treatment including other medications conjunction with the insulin. 5 to 10% cases this for type 1. Type 2 predominantly the various of medications are available to treatment manage of blood glucose. Often require insulins, often delay the longer and recommended. The most promptly affected for the patients type 2 diabetics for the 90 to 95% in cases. Progressive conditions involving changes in 8 different organs, body including the pancreas, liver, muscle and kidney. In onset of occurs at any stages, this increases the risk of eating disorders and can lead to exhaustion, frustrations and fears with causes of this diabetic. Moreover, compared to the type 1 and type 2, rates of onset of increasing among the general populations worldwide, they are affected for this both. Next. The second one is gestational diabetic. Gestational, we know about this pregnancy time during this affected for this gestational diabetics. This woman's possibly there are children also risk of the type 2 diabetic in futures. In gestation diabetes diagnosed through the parental screenings rather than through the symptoms. Next. In my research, the valeric acids and cephalosporin diphenhydramide we are using for these drugs. In substance 1, cephalosporin, Micropharmas C8, H15, N3O7. We are utilizing 45 milligram per body weight is a buffer using for this intraperitoneal injection to the rat for 1 ml. This compound nature is a glucosamine, nitrogen, urea. The second one is nicotinamide to suppress for this activities nations C6H6N2O. This is 110 milligram per body weight is normal salines to distribute with it, along with the 15 minutes after the cephalosporin, simultaneously nicotinamide give them 1 ml for intraperitoneal injections. This is a natural compound is a pyridine carboxamide. In third substance, the standard drug we are using for this consumer of metformin and different type of drugs. In my studies, it is for the diphenhydramide. C23, H28, chlorine, N3, oxygen 5+. This dosage I study for the 600 milligram per kg body weight in distilled water. Intragastrically, 1 ml. This nature of the compound is sulfonyl urea. Hello, next. The vehicle we are using for this simultaneously for my drug, using for dimethyl sulfonyl sulfoxide. It's dissolved in the water, plastic with 1 ml. This compound nature is a organosulfide. And then valeric acid. This valeric acid name is 3,4-dimethoxybenzoic acid. This is a benzoic compound. I do using the dosage for 40 milligram per body weight. 1 ml of DMSO is a vehicle dissolving this compound as per the body weight. Intragastrically, this is using for the phenolic compound. Next, this natural compounds found in the fruits and vegetables. This is a behavior of the antioxidant and anti-inflammatory, blood pressures and lowering effects. The protective against the hypertensions associated with the cardiovascular renal disease. This plant is from Vitis vinifera. This is also activity of valeric acid is an antimicrobial activities. Next. Next bill. This administration of cephalosporin, this is the main cause of beta cells inducing for this diabetic to reduce these insulin secretions. There's some protocols preparations for this cephalosporin. The main reason for this maintain for the pH stabilities along with this nicotinamide derivatives of vitamin B3. This is vitamin B3 we know what the niacin. Reduce the streptococcus such as the effect of the B cells through various mechanisms, inhibit the free radicals, inhibit for this PAPRP produced cytokinin induced NHC cells to express provide with NAT+, enhancing B cells regeneration, inhibit apoptosis. This is DA methylation, particularly in pancreatic B cells, nicotinamide administration intraperitoneal normal cell lines. This is for these activities of insulin dependent. Next one. Yeah, this is an experimental group, has 6 groups. First group is a controls, second is a normal rats and valeric acid 40 mg per body weight for 30 days. We are grouping for Arham. Then group 3 is a diabetic control rats. Injected, same things we are previously know about this, how this administration of streptozotocin. The same thing after 15 minutes after intraperitoneal administration with the nicotinamide to maintain for that type 2 diabetics. The main thing is the group 2 for this type 2 diabetic to studies for this along with the streptozotocin. The group diabetic rats along with the administration with the 40 mg milligram per body weight of valeric acids. Orally, we have consumed for the 30 days after. To maintain for the each and every days. Next one, group 5, diabetic rats is administration with a diphenhydramide. The same 600 mg per body weight orally for a period of 30 days. Yeah, then the study about before the treatment, first initially we find out the blood glucose, the control around this 89 and drug means bariatric acid 40 mg per body weight 153. When compared to this one, this slightly increased in this normal, and this drug to reduce these diabetic activities. In this diabetic control, they are increased in the diabetics range. When compared to the standard drug of glycine and diabetic range is nearer about the drug bariatric acids. In fasting insulin, the same relevant for this standard drugs in this diabetic for this bariatric acids and compared to these diabetic controls and for these standard drugs. An effect of glucose and insulin and glycogens. The diabetic source markedly evolved with the levels of glucose compared to the non-diabetic control. However, upon the administration of bariatric acid, the level of effectively reduced, indicating the ability of reduced blood glucose insulin level in diabetic controls. This is a previous one before treatment, after treatments. This is insulin.
Excuse me, sir. Last 2 minutes.
2 minutes. Okay. Insulin and glycogens, these are the two hormones, so non-regulative of the blood glucose levels. To increase this blood glucose is a glucogen from alpha cells. So in beta cells, to decrease blood glucose level for depend on this insulin. The most predominantly we have studied about the lipid profile, total cholesterol, triglycerides, high density lipid profile, low density lipid profile, VLDL and non-factors. In this studies, control along with the drug means bariatric acid by drug, then diabetic and diabetic rats with drugs and standard drugs. The impact of lipid profile diabetic rats exhibits the drastic changes in the plasma lipid profile, likely contributes to the progression of diabetic complications. Bariatric acid result in this reverse of alterations indicated potential to mitigate dyslipidemia associated with diabetics.
Excuse me, sir. Can you move for the conclusion?
Yes. In my drug for anti-lipidemia to reduce our processing and reduce the glucose activities of these drugs. So these are main conclusions for the drug to induce further bariatric acids. Thank you.
So I would like to know overall outcome of your work, simply in 2 lines.
The main thing, this is a polylipidemia to activities to reduce this glucogen and to enhance for the activation of diabetics. This my drug happens.
So is it useful for both kind of diabetic type 1 and type 2?
It's type 2 only.
Only for type 2?
Yes.
Good evening, sir.
Yeah, good evening, sir.
So how did you analyze your data, sir?
For ANOVA SPSS data. This data were analysis.
But I don't see any significance in your
The significance is not included.
No, it is not at all mentioned. We could see only standard deviation in your chart.
SPS ANOVA, we have done it for there.
Yep, you might have used.
Yes, sir.
I'm not telling you that you didn't
Yes,
use it in your chart.
Yeah.
I don't see any comparisons.
Yep.
Without comparisons, without significance, how did you come about results and conclusion?
Already we done it, but this slide is some mistakes.
Okay, right. And why did you do in silico after your in vivo studies?
Previously, we find out the in silico, then we go to the animal work.
But you presented animal work first and then in vitro.
Of course.
It is not logical.
Yes, it's logical. I understood, but previously we done it in silico, and now this.
You could have done in any order, but you could have presented in a
Exactly.
And what is the reason for choosing valeric acid?
This is a main source in vegetables. So both of the people in our diabetic peoples are consuming of the vegetables. The presence more prominently in valeric acids. So that's why we are find out this anti-diabetic effect in this compound.
Thank you, sir.
Thank you.
Okay, thank you, sir.
Thank you, sir. Next, I would like to call Tanima Deknath Sarkar from Annamalai University.
Good evening to all. I am Tanima Deknath Sarkar, Annamalai University, and my PPT is gene silencing influencing gene expressions as a multifactor regulatory and recovery molecular mechanism and the hepatoprotective functions, which is the focus of the Padina gymnospora. Sir. Okay. Come. This one. No. This one. Okay. Thank you. The gene silencing functions as a regulatory mechanism providing the negative feedback and manage both the gene expressions and the metabolic process over the lifespan of an organism. This molecular process enter the subsections of the particular genes and could have the develop of the protective mechanism against the various and the foreign nucleic acid. The operations of the gene expressions encompass 4 key steps. One is the RNA expressions, promoter analysis, protein expressions, and the post-translations, the modifications. Within the framework of the gene silencing transcriptions and the translations represented the 2 distinct levels. Histone modifications contribute to gene silencing at the transcriptional level. Methylations of the silencing promoter within the nucleic results in decreased RNA synthesis. On the other hand, the translations level in the cytoplasm active the methylations of the coding region engage in the sequence, specifically RNA degradation through the formations of the double stranded RNA and the genomic, the imprinting in mutations, positions effect, RNA, direct DNA methylations, transpons, the silencing and the transform silencing are the 4 methods, and these are the transcriptional, RNAi, nonsense mediated decay, and are the 2 methods. These are the post. And Padina gymnospora is my work is a brown main algae and the functional bioactive molecule and the low non-toxic properties providing the protections against the non-alcoholic liver disease. Gene silencing serve as a regulatory mechanism through the negative feedback influencing gene expressions to determine the cell fate. Additionally, it plays a pivotal role in governing both the metabolism and the gene the entire lifespan of the organism. On this the introductions I wrote the who discovered this one. There is this work was done the exclusive the fundamental principles of the siRNA structure and the mechanism of the RNA infections lying down the work. Professor Andrew Fire and the Carriage Mallow who were awarded for this reason in a Nobel Prize 2006. Then this is the continuations, the how to gene silencing is going on. The transcriptions of gene silencing TGS. So this is the siRNA mediated pathway and the RNA mediated pathway, how it is running. Here is this, I am showing here. Transcriptional gene silencing, the outcome of the histone modifications of the establishment, the heterochromatin in the environment, the surrounding gene redending at the incredible, the transcription, the mechanisms, including RNA polymerase and the transcription factors. So there is a different way the chemical modifications, the histone induce the changes, the packing state of the DNA, influencing the transitions between the urochromatin and the heterochromatin. Histone is acetylations for the instance of the and can the destabilized chromatin structure by the modification of the chain chromatin with the chromatins. This is the method of the gene silencing, genomic imprinting, permutations, positions effect, RNA direct methylations, transforms silencing and the transient silencing. This is the way how to genes, somatic tissues and the parentally and the maternally it is going to work. There is some definitions which I told, the genomic imprinting, in positions effect, everything is I have given. This is the picture of the paramutations. This is the way the permutations is going on. These are RNA directed DNA methylations. How to occur this one I have showing here. Transient silencing. This the way the transient silencing is the continuing process. Okay, last is the post transcription gene is a silencing. This is the two way. One is the RNA inference and the nonsense mediated decay. The molecular mechanism of the post transcription gene silencing, how to occur this imaging is the showing. RNA inference, how to do the work on RNA inference. This is the way the RNA inference is the working. In p-miRNA, then it is going to the miRNA, and after that both siRNA and shRNA, RNA-RISC complex, and the enzyme cleavage and the target mRNA and cleavage manufacture of the mRNA. This is the continuation, the working process of the RNA. This is the PCR test. I have to done the PCR test because there is a long paper. I am giving in a very short way, focusing only the gene silencing, not more than the gene expression. This is the RNAi pathway. RNAi pathway, how to occur this one, the nucleus, and after that the mRNA cleavage in cytoplasm, and this is the way the RNAi pathway and the regulatory expressions how to do. Then I have to write only the gene expression analysis is a different physically DNA as the consist of the single chain and the exhibit in the greater chemical reactivity in contrast to the RNA, which is the conflict to the nucleus. RNA is a more mobile with the various stabilities of the performing distinct functions. RNA molecules is effective, read the instructions, encode in the DNA and then and the initiated the necessary process to translate them into the actions. The communications of the process is result in certain the functional product like a protein is termed gene expression, and the gene expressions how to work the influencing gene, the factor, time of the sample.
Excuse me, ma'am. Over here. Ma'am, your last 2 minutes, ma'am.
Oh yes, finish. Time of collections varies over the 24 hours, environmental and the inherited. My work, the Padina gymnospora is a brown algae, accelerating in the bioactive compound, and the medicinal antibiotic is a therapeutic, and this is the working is a qualitative analysis and the high performance in a layer of the chromatographics we have done with PCR test. And last conclusions what I want to tell, gene silencing in the emerges as a crucial regulatory mechanism. Gene expressions can the metabolic process throughout the organism in the lifespan, And this is the vice versa, histone modifications and the RNA interference represent the pivotal mechanism in the gene silencing impacting the transcriptions and the translations process. Various methods such as the genomic imprinting is there, and it is generally Padina gymnospora highlight in the potential the therapeutic applications of the understanding the gene silencing in the combating disease like a non-alcoholic life disease. Liver disease. Overall, the gene silencing underscore the significance in the heuristic, the cellular process in the influencing the cellular fate and the serving as a fundamental regulatory mechanism of a biology. This is the conclusions, and I thanks to the Arun Selvan, Mohandi, Jan Albuvani, and my guide, Dr. M. Mudlingam, sir. Okay, sir. Thank you.
In your work, you said it's active against all kinds of non-alcoholic liver diseases. Isn't it your conclusion?
Yes.
So, can you specify any type of liver diseases which is working?
Ma'am, beg your pardon, please.
You said that non-alcoholic liver diseases, it is silencing the genes.
Exactly.
So, what kind of non-alcoholic liver diseases? All, that's why I'm asking, it's workout in all liver diseases or any specific liver diseases?
Any liver diseases.
Any liver diseases, even cirrhosis?
Yes, liver cirrhosis. It is effect.
Acute or chronic?
Yes, ma'am.
Acute or chronic?
Acute, chronic, both.
Both.
Yes, ma'am.
Ma'am, is this a review paper or research paper?
It is my research paper.
I didn't see any results in your presentation.
No,
I didn't read it.
here is a PCR test, whatever the full data, I couldn't give because they are telling the only the 15 slides. How should I maintain gene silencing and the gene expression is the word.
I don't see any results at all.
This is I only
It would take only one slide to represent the results of your gene expression studies. You could have made a table with increase in fold expression or decrease in fold expression.
But, sir, I just only the last paragraph I wrote in a Padina gymnospora what is working. The last.
Did you treat your extract in animals?
Yes.
I don't see any animal group.
Oh, no. Here I am not giving that. I am extremely sorry because this is the big things I am not controlling in a small way.
Okay.
That's it. I'm sorry for that.
Yeah. What is your actual budget, ma'am? Your project seems very costly.
Yes.
What is your actual budget, ma'am, if you don't mind?
No, I am trying.
Okay, ma'am. Thank you.
Okay, expensive.
Today's session got over. I thank everyone for your active participation throughout the day. Good evening and see you all bright and early tomorrow at 10 a.m.